Stephen Burley. I'm the founding director of the Institute and also the director of the RCSP Protein Data Bank. The Institute today is very pleased to be hosting a virtual crash course that's being put on by the Department of Energy Knowledge Base, KBase, and the RCSP Protein Data Bank, RCSP PDB. And our focus today is going to be on using KBase to access PDB structures and computed structure models that are coming from artificial intelligence and machine learning. I want to deliver a special welcome to those of you who are joining us from around the United States and around the world. It's a great pleasure to be able to host these crash courses virtually and have so many individuals be able to join us literally from around the globe. The Zoom today will be recorded and it will be posted on the RCSP PDB YouTube channel. The presentation PDFs will also be made available to participants via the exit survey, and I'm going to provide the URL at the end of today's event. Before I introduce my co-organizer, Chris Henry from Argonne National Lab and a member of the KBase team, I just wanted to provide a little bit more information about what we mean when we use the term computed structure model, and make a distinction between the kind of information that's stored in the protein data bank and the models that are being generated by artificial intelligence machine learning methods. So on the left of this graphic, which came from our New England Journal of Medicine paper that was published late last year, we contrast the workflows for experimental structural studies and computed structure model calculation. Both approaches make extensive use of genomic data and the 3D structural data that's stored in the protein data bank. On the experimental side, the sample has to actually be generated, purified, and then prepared for measurement. And a structure determination process involving extensive computational resources is carried out, resulting in an experimental structure. These experimental structures, particularly for eukaryotic proteins, are typically only partial in length. They have either very few or no regions of the structure where the confidence is low. And functional ligands are often present. So the example that's shown here is of a KH domain, a fraction of a portion of the NOVA2 RNA binding protein interacting with its RNA target, this this RNA hairpin. The information that's published from crystallography, NMR spectroscopy, and electron microscopy ends up in the protein, being made public through the protein data bank, and the URL for the RCSP PDB research-focused website is rcsp.org. In contrast, we see computed structure models, again starting with genomic data and 3D structure data from the PDB, going through a complex computational process that results in computed structure models that are typically a full length, but for eukaryotic proteins in particular, they often contain many low or very low confidence regions and no functional ligands. As a practical matter, many of the human proteins, many of the plant proteins, so for which there are computed structure models, look like beads on a string or spaghetti and meatballs. The beads or the meatballs being the well-defined globular domains, such as this KH domain, which corresponds to this part of the PDB structure on the left-hand side, and then two other KH domains, all three of which were predicted by AlphaFold 2 at high confidence, but about 70% of the polypeptide chain could not be predicted with high confidence, and it's probably intrinsically disordered in the absence of other protein partners, and is not actually amenable to study by X-ray crystallography methods. And I know this from experience because my before determining this co-crystal structure, my team at Rockefeller University, where we were part of the Howard Hughes Medical Institute, attempted to express, purify, and crystallize the full-length NOVA2 protein consisting of 3 KH domains and then all of this spaghetti. And we were never able to crystallize the full-length protein because the spaghetti is mobile and gets in the way of forming good crystals. So the information to some extent is complementary, and that is that underpins the argument that we have, the rationale that we have, motivation that we have at RCSP Protein Data Bank for delivering both experimental structures and computed structure models together. And the 4 reasons are enumerated here. With computed structure models, we've got 3D structure data for all model organism proteomes. As I described with the NOVA2 case, the computed structure models cover entire polypeptide chains, unlike many PDB structures. The computed structure models have been formatted in such a way that they can be delivered alongside the PDB structures using the full functionality of the rcsp.org website, and you'll see that exercised extensively. We'll cover searching, visualization, and analyses. And I want to be clear that when we present this information on the rcsp.org website, the computed structure models are clearly distinguished from the experimental structures and clearly identified as to their provenance and accuracy. Our overarching goal in providing this one-stop shop for access to both computed structure models and experimental structures was to provide better service for the more than 99% of our web portal users who are not experts in structural biology. They want information across entire model organism proteomes, and are also interested in the parts of the structure that may not be accessible through one or more of the experimental techniques supported by the Protein Data Bank. So with that, I'm going to turn the podium over to my co-organizer, Chris Henry from KBase at Argonne National Laboratory. And then we'll work through a series of presentations from both KBase and RCSP Protein Data Bank team members. We'll take a short break at 3 p.m. Eastern time for 15 minutes, just to allow people to catch up on email, use the bathroom, etc. And then we'll conclude with 3 additional talks. And then I'll sum up at the end, and we'll be finished before 5 p.m. So with that, I am going to stop sharing. I'm going to thank you in advance for your attention, and leave you in the very capable hands of Chris Henry. Take it away, please, Chris. Thank you.
All right. Thank you, Stephen, for the introduction. So I'm going to be presenting. I'm going to steal probably a little bit of Jonica's time, so this session might run a little late, but I'll also be covering this Arabidopsis case study. So I'm going to go over the crash course objectives. I'm going to give you a little introduction to KBase for those who don't know that platform very well, and then I'll get into this particular case study to get things rolling. So the course objectives here, really, a big thing is to teach those who are new to either PDB or KBase that are attending this workshop a little bit more about these platforms. So hopefully, if you're like a KBase wizard, you're going to learn a lot about PDB today. If you're a PDB wizard, you're going to learn a lot about KBase today. And we want to highlight, in particular, some new tools we've added to KBase to enhance connections to PDB. And we want to demonstrate how these tools can be applied to discover new functions and genes in plants and microbial genomes. Generally, we want to improve your knowledge and key tools in KBase, so modeling, genomics, annotation, and in PDB, like MolStar, structural query, and structure alignment. And importantly, we want to get your feedback on the tools we've built so far, and your suggestions on what we should build next. So the course outline, I'm not going to get into this because I think Stephen already covered this quite well, but I'll cover an Arabidopsis case study. Jonica will cover a microbial case study. Dennis will talk about experimental structure analysis in PDB, particularly focusing on Arabidopsis rhizostome. Brenda will talk about exploring tools and data in PDB to aid in function discovery, picking up from where Jonica and I leave off. And then Shuchi will do a detailed tutorial on visualizing structured data in KBase. QZ will talk about structured tools in KBase, and then I'll take off from there, just exploring any various kinds of information that can be gained from the PDB tools in KBase and discussing future plans. And I think Stephen will do some more future plans in the concluding remarks. So with that, I wanted to do a very brief introduction to the KBase platform, if you're not familiar with it. KBase is an integrated platform for aggregating and sharing tools and data in order to collaboratively solve scientific problems. It is somewhat focused on the BER space, but that doesn't mean that anybody can use KBase. It's free for anybody to use, even commercial use. It's completely free and open platform. To achieve its mission, KBase has a capacity to do a lot of data management. So currently there's unlimited data storage. That may change at some point, depending on how the data, ecology evolves here. But we can import data into KBase by drag and drop into this import menu, and from Globus and from a variety of other online cloud sources. And you can import a variety of data types. And a big thing that differentiates KBase from a platform like Galaxy is we don't work in files, we work in objects, which is nice because all the files get converted into a standard format, and then that's shared by all the tools. So if you have a tool, you don't have GFF and GenBank tools in KBase, you have genome tools in KBase. We also have a wide range of data analyses, increasingly coming from third-party developers. So currently KBase is up to 280 different analysis applications covering read processing, assembly, annotation, alignment, comparative genomics, metabolic modeling, and omics analysis. And we support collaboration. That's a big part of the mission for KBase. It's where people can take their data, keep it private, and work with it and analyze it and do your science in collaboration with specific other users. You can share data with specific other users. And then when you're ready to publish, you can make your data public, and even get a DOI for your studies. All this is shared in the form of narratives, which are Jupyter notebooks with our apps in them. And users can also gather their narratives into, users and common organizations can gather their narratives and share these on organization pages in KBase. And these are used for a wide range of purposes. We have science focus areas like projects and DOE that have an organization page. We have an organization page for educators, for everybody who uses KBase as a teaching tool. We have organization pages for many classes at the high school and college level that they actually are using KBase to teach a class. And we've got organizations that cover data sets like large MAG and SAG collections from various publications. And what's a narrative? So a narrative is it's built on Jupyter notebooks. It's a notebook interface, but it's a little different from Jupyter if you're familiar with Jupyter. In one case, you have an object-oriented data panel with all of your various data objects for analysis here. You have a point-and-click app panel for your various application analyses, and you point and click on these and it adds the app to your notebook, and then you run the app and you see the results. And the notebook itself has all of your analyses, but also it has capacity for markdown cells as shown here. And you've got various buttons to share your notebook with other people and to do more markdown cells and code and even code cells. You can programmatically interface with the KBase job engine in these notebooks. We've got documentation, numerous tutorial narratives from kbase.us, and we've got a YouTube channel, as does PDB, with videos of many of our workshops, and we'll be posting links to the video of this workshop there, so you can look for that. If you missed part of the workshop, you'll be able to see it in these video posts at some other point. So why link KBase and PDB? So there's an enormous and rapidly growing synergy between these two platforms. KBase has extensive genomics data, but one thing I want to emphasize, because it's kind of differentiating from other platforms, is a lot of it's not reference data. This is like people's specific isolates in their labs, which might be very, very similar to a reference genome, but it's going to have differences. So the KBase is kind of a home for that. You can put your non-reference data in and then contextualize your data in the context of reference data, but also other people's non-reference data. For example, there's a lot of genomes that only exist in non-reference form. They're isolates. They've never been isolated in pure culture. They're from microbiomes. And the only way you'll see it is if we can compare your non-reference data with other people's non-reference data. And that's one of the things KBase is meant to facilitate. We have numerous workflows to integrate multi-omics data, comparative genomics, and to predict and understand protein function. And now with advances in protein folding, now you can take this data in KBase. You can't fold it yet in KBase. We're hoping to get there at some point, but you can take your non-reference gene sequences in KBase, export them, fold them in AlphaFold, load those structures into KBase, but then compare with PDB. So this is where PDB shines, is they've got this enormous database of experimental structures, plus they're now aggregating computational structures. So KBase is potentially an avenue to eventually be able to publish computational structures in PDB, register those in PDB, although we're not doing that yet. But also it provides you an ability to quickly compare your genes and your computational structures in KBase to the experimental data in PDB. And as Stephen just spent a long time explaining, it's very important that the experimental structures offer an enormous amount of information. The computational structures alone, they really don't stand alone. You need those experimental structures too. And of course, PDB has a growing body of just general structure tools and capabilities and data that is valuable. So by linking these two environments, we synergize, and the beautiful thing is doing this in an environment of rapid protein folding just makes that synergy all the stronger. So with that, I'm going to jump to a little case study to get things started. I'm going to focus on Arabidopsis for my case study, and then Jonica will cover a different story in the microbial space. So I'm going to hop out of my presentation and jump to my KBase narrative, which is here. I'm just going to make this full screen as much as possible. I'll check the chat. One thing I'll say is you'll notice this message periodically streaming in the chat. We have an online Google Doc. I think I have it linked here. Where we post various information about the sessions that we're covering, including the key sequences and gene IDs and links to PDB. But also there's this Q&A section. So please put your questions here. And other panelists will monitor this as the workshop proceeds, and will provide answers as we go. So you don't have to wait until I finish to get your questions answered. We'll answer as we go. So just to post a question, just copy and paste, just as was done by Herman here. Just copy and paste the Q and we'll add an A. All right. Awesome. So let's go to this narrative. So this is covering a little metabolic mystery in plants. We're going to go back in time. This is a lovely thing to do in science because then we know what the answer is ultimately. So we can't be wrong. Well, we're going to go back in time to 2012 when this paper was published, Plant B, and there's a link to the paper here in the narrative, and there's a link to the narrative in the Google Doc. And if you do need to have a KBase account to get to this narrative, but I will post in that Google Doc a HTML version of this narrative right after I finish speaking, so that you can get to that without a KBase account. But this is a figure from that paper, and what it's showing, this paper covers in general mysteries of B vitamin biosynthesis in plants at the time, 2012. And what this is a figure from that paper showing riboflavin biosynthesis specifically, and highlighting the fact that the PRR step is unknown in riboflavin biosynthesis for plants at the time of 2012. So you can look at that. So it's just highlighting that this is unknown. And this was done by a rigorous literature search to compile all these mysteries by plant researchers. But the first question that occurs was, how does KBase tell us that this mystery exists? And so there's an app in KBase that lets you reconstruct the metabolism of a plant. So you just give it your plant genome. It has to be annotated with an app that's linked in this description here. You can see the link here, and the annotate plant enzymes with OrthoFinder. But once you've annotated a plant genome, you can build a model using this modeling app. And it will provide you with all of the reactions, but the model does, this is a list of all the reactions, their compartmentation, their function, and the genes in the plant that have been mapped to those reactions. And you'll notice there are reactions without genes. So in this case, the model is doing something we call gap filling, saying, I know this reaction, it has to be here to make this organism work. This metabolism doesn't work if you have this hole in this pathway. You have to fill this hole in this pathway. And what we'll see with this 2012 version of the Arabidopsis annotation is if we look up our step in Arabidopsis biosynthesis, we look up 1.1.1.193, our EC number, we see there's no genes. So this is KBase's way of telling you what this paper is telling you in a much more elegant, visually compelling way, that this is a mystery that needs to be solved. We don't know what Arabidopsis gene does this function. We know it has to be there, but we don't know what gene does this function in Arabidopsis. You get a nice view of the model, and as well, this is the model viewer for KBase. It shows all the reactions with the compartmentation, the genes. The genes are clickable, so you can bring up the gene information. It shows the compounds with formula and charges, the compartments. You can see the plant models are fully compartmentalized. And the nice thing is this model is also fully functional. So you can run flux balance analysis to predict the flux through the pathways in this model. And just by specifying the model in the media, in this case, the media is autotrophic. So we're predicting the growth of these cells on just CO2 and light. So they have to make their vitamins. They can't just import it from the environment. And the nice thing is we can visualize, if you're a pathways person and you want to see the hole in the pathway visually, we've got a genome-wide, a genome scale map of plant metabolism with the fluxes from FBA painted on it. So you can see all of the reactions, and this is fully compartmentalized, although we don't show the compartments on the map because it's a mess. Because you've got pathways that go in and out of different compartments. And again, you've got the genes listed if there is one for a reaction. And what this is showing is that for this particular pathway, this is just another way of showing that gap in the pathway. Like here is your missing step with the no, it has flux, but it has no gene. So yet another way that KBase is showing that there's this hole in the pathway. So we accept that this is a mystery we want to solve. How do we solve it? So we can annotate this genome in a variety of ways, and that could be an approach. What I'm going to highlight is using the PDB apps to attack this mystery. And the PDB apps are special because it brings all of the rich metadata and information that PDB has to bear on your problem. What this first app I'm going to show you is, it's called Import PDB Metadata into a KBase Genome. And this app, it gives you an ability to take an entire genome, every gene in the genome, and query PDB with it. And then pull in all the metadata in PDB that relates to the genes in that genome into that genome. So these are going to be orthology hits, and sometimes they'll be exact hits. I think we'll see an example of that. But these will generally be orthology-based hits. And the key thing to recognize here is this app, by necessity, because it's doing a whole genome, it's doing every gene in your genome, it's meant to be exploratory. So it's a broad but shallow dive into PDB. I'm only keeping the best 2 EC numbers, and I'm only keeping the best PDB hit for each EC number that I keep. Here for Arabidopsis, you can sort of generally see the PDB coverage. So you get, and this you can set the EC number, the E value filters and the identity filters. But for the particular filters I chose, which I think are 10 to the -10 E value, you've got 14,000 hits. And that's only keeping the best hits. So that's roughly 7 to 14,000 genes. And now we can look to see if we have any hits that are relevant to our metabolic mystery, just by searching for the EC number. And what we see is that, yes, indeed, we have two genes in the Arabidopsis genome that have hits in PDB that link to this EC number. Of course, I can probably annotate this genome with a variety of tools and also find this EC number. But the nice thing is we also now have a link to PDB. So I can click on this link to PDB and get a picture of the structure that I'm looking at. I can see the method that the structure is obtained, the strands. This is going to be important later, but you can see how complete your complexes are. And you'll notice here, you've got very different hits on the strands for the two different genes. And we'll see that to a greater extent later. You can see the taxonomy of the hit and the name of the function itself, the co-crystallized compounds. And the nice thing is having that broadly, and I'll talk about this more later for the genome, is really useful. The links to PDB and the links to the papers. So a lot of really rich information. But now I mentioned broad but shallow. So we've got these two genes that are interesting and we want to dive into more. Maybe one of these is the answer to our metabolic mystery. And so we have a separate app, Query RCSB Databases for protein structures. And for that, you can put in the protein sequence of our genes of interest with filters and then see the results. And now you get not a broad but shallow, but a deep dive into all of the hits for each of these genes. And the interesting thing that you can see from this is, again, if we look for our EC number and we look for coli, we see there's a gene in E. coli that hits both of these structures. So like now we've got a single structure. It does the function we were looking for, and it hits both of the genes in some way. And at this point, I'm going to hand the story over to Brenda when she talks. She'll take it from here, like what can you do? So you can link out to PDB with these structures. But the other nice thing is that PDB also has computational structures for all of Arabidopsis. So we have computational structures for our two genes of interest. And so we can dive into those. And all of that is linked with all the sequences in the Google Doc. So we can chase this down further. So we'll leave it at that. And then Brenda will see what PDB has to say about whether or not these two genes do this particular EC number. So with that, I'm going to hand things over to Jonica, who will talk about a story in microbes.
Thank you, Chris. So I'm pleased to introduce Jonica Dirisinji, who's in the KBase team at Argonne National Lab. Thank you very much for agreeing to speak today, Jonica.
Yep. Thank you, Stephen, for the kind introduction. So again, thank you for joining us this afternoon. To properly orient all of you to the rest of the presentation, I'm going to present a scientific workflow and then discuss how we effectively use the RCSB data to achieve our scientific goals in this specific use case. So the scientific problem that I'm discussing here is that we know that there are so many microbial transformations yet to be characterized. So how should we computationally predict these potential gene candidates for these novel enzymatic transformations, and then how we can effectively use this RCSB data in identifying these high confidence gene candidates that was predicted from the computational pipeline. So the specific scientific example is that the scientists at Argonne had this experimental strain, Micrococcus luteus, through phenotypic data that the strain can grow on pyridine. So, but the pathway wasn't explained until very recently. So the organism can grow, but the degradation pathway is unknown. So in this workflow that I'm going to show that how we use the KBase functional discovery pipeline and also the knowledge that derives from RCSB, there's a wealth of knowledge that we can get from this RCSB data and identify this potential pyridine degradation pathway. So very quickly, so this will show the KBase functional discovery pipeline apps and also the apps that fetch the PDB data, RCSB data. And so I'll discuss this in a minute. But what I want to show here is that what this graphic shows here that, so when you start with a genome like Micrococcus luteus that has about 4,700 genes, how we can filter the genes that may play a role in this novel degradation pathway. So we use these genomes that has about 4,700 genes, and when we build construct metabolic models, that you can see from 4,700 genes, it's reduced to about 1,100 genes. So the about 3,500 genes get put aside, because now we are talking about all the metabolic genes. So we use these tools and we use the structure analysis, and then we get about few genes that we can ask experimentalists to verify in the laboratory environment and to identify and characterize these enzymes. So with that, I will go to the narrative workflow. So I have a link to this narrative workflow. I will also put a link to a HTML version of this narrative workflow. So if you don't have a KBase account, you can still access this narrative. I'm going to put that the HTML version of this narrative into the chat. And so what we are doing here is our starting point is the microbial genome. So we have a microbial genome which is the Micrococcus luteus. The first thing that we want to find out is, okay, so we don't know there's a pyridine degradation pathway. Can we annotate these genomes with multiple annotation algorithms and try to find what functions are present in this genome? So here we annotate in this workflow with multiple annotation algorithms like RAST, PROC, and DRAM. And when we annotate this genome, so you get these annotated genomes, and that you can browse that annotated genome in the KBase narrative. And you can search for functions. So here that we can search for function like, pyruvate synthase. And can we find anything to do with pyridine? Any functions, anything to do with pyridine? We get some hits that related to tetrahydropyridine, but this gives me very little information. I don't see any pyridine degradation pathway related genes in this genome. So from here, we want to construct a metabolic model. So what we are doing here is we construct metabolic model based on the annotations that was generated above. So we map these annotations to biochemical reactions. So I have most of my slide transferred here into the narrative so I can save some time. You also have my slides as a reference to go over. And we construct this metabolic model. So once you construct a metabolic model, now if you see the output of this metabolic model, what you see is a bunch of biochemical reactions. Now, note here that each biochemical reaction here is mapped to a specific gene. So this is the biochemical reaction that represent by this particular gene. And it also gives the functions of these biochemical reactions. And I have added documentation here that what each of these tabs represent. And I also have a link to tutorial. If you are less familiar with the metabolic modeling, there's a nice tutorial here that you can link out and learn more about that. So, okay, so now we have a metabolic model. And if I simulate this metabolic model to grow in the presence of pyridine, and obviously I won't get any growth because there's no pathway for the pyridine degradation. Now, what do we do? So we use this Pickaxe app to generate potential utilization routes for pyridine, potential new utilization routes for pyridine, since there's no known reactions in the known biochemistry. So this tool uses a set of general reaction rules, which are curated from known biochemistry, and generate novel compounds and reactions based on the compound of interest. So in this graphic, what it shows here is the pyridine molecule. And based on known biochemistry rules, this expand this and generate novel compounds and reactions. So this shows the generation for a single generation. So this in this study, in this specific workflow, we have used 3 generations. So each of these compounds will again expand into novel compounds and reactions. So it's worthwhile to mention that if we have additional phenotypic data, just like metabolomics, we can map the metabolomics into these newly generated compounds based on the molecular weight. But in this specific user case, we don't have the metabolomics, we don't talk about it. So now we are doing this now because there's no pyridine degradation pathway. We need to identify a potential route. Can we identify a new potential route, new pathway, computationally predicted pathway to degrade pyridine? So the output of PICAX now shows a bunch of reactions. Now it's generated, and it's also this is also worth to note here that with these newly generated reactions, it gives this partial EC numbers. So here for every reaction, it gives this partial EC numbers, which become very handy down in the pipeline. And so the first 3 digits are what's important here that show the partial EC numbers. If I'm right, I think the next 2 digits represents the specific biochemical role and 159th reaction for the specific biochemical role. So let's focus on the first 3 digits here, which provide the partial EC number. So now we have a bunch of reactions that potentially can degrade, show degradation of pyridine. Now let's try to see whether we can use these newly generated reactions and then integrate that reactions into the metabolic model. Now you see here that it gives me about 7,000 reactions from this PICAX output. Now, how do we pick the specific reactions that play a role in this pyridine degradation? So what's happening here is that, in summary, so we have this model, metabolic model constructed from genomic annotations that has gaps for the pyridine degradation pathway, and we use the PICAX app to generate novel compounds and reactions for the pyridine degradation. And we try to integrate this set of novel reactions in combination with known reaction and try to fill this gap so we can potentially have a pyridine degradation pathway integrated into this model. So that's what this gap fill metabolic model app do. So when we run the gap fill metabolic model app, it integrates this novel set of reactions. So the output of the gap fill metabolic app here shows, now we can see these reactions that has the pyridine degradation reactions, like for instance here, that pyridine get converted to 3-hydroxypyridine and 3-hydroxypyridine get converted to 2,5-dihydroxypyridine. Very importantly, that we can also see the partial EC numbers. So these are the very first clues that we can search in the genome what potential genes may play a role for these particular reactions. Also note that there is no genes associated with this reaction, and that's obvious, right? So these are novel reactions that was predicted, and we don't know what the gene, what genes are playing a role that representing this biochemistry. Okay, so now we have a potential route that was computationally predicted for pyridine degradation. And by the way, I also should note that known reactions have these numbers that are RxN ID, and these weird looking reaction IDs are the reactions that was introduced from the PICAX output. Okay. Now the next step is, now we have a potential route, and let's simulate in silico. So let's try to simulate grow the Micrococcus luteus model in the presence of pyridine minimal media, and using flux balance analysis under aerobic conditions. And so we have an app run flux balance analysis, and I have provide some documentation here how you can produce a pyridine minimal media. So when you run the flux balance analysis, now we can also see the fluxes on those newly introduced new pyridine degradation reactions, and that's good. So in summary, what we have done here is that now we can simulate the potential pyridine degradation pathway, and we use a media, a pyridine minimal media. So the sole carbon source is pyridine, and it simulates how that the cell uses pyridine as the sole carbon source and produce its biomass, or it can grow in the presence of pyridine. So this also showing here in a pathway map. So we can open this in a separate window so we can see this clearly. So here's the cell membrane that pyridine comes into the cell and converts to 3-hydroxypyridine and then to 2,5-dihydroxypyridine, get converted to malleate, and then get fed into the TCA cycle via fumarate. So this is the proposed new pathway. So now we have a pathway in terms of biochemistry. Great. Now, the next question is now, how do we identify potential gene candidates? So this is much harder problem. We have a pathway in terms of biochemistry. Now, identifying potential gene candidates it's more challenging question. So we have done the differential expression analysis on this genome. So the differential expression analysis was generated based on glucose versus pyridine, and so this will give us more evidence that we can eliminate some of the genes that are not expressed under the pyridine. So let's go back really quickly to this to the model that the partial EC numbers for this newly introduced reaction, which is 1,14,13, and I'm coming back here. So in the genome, if I search for 1,14,13, I find about 30 genes. This number is a bit off. It's an artifact because some genes are represented twice here with the different annotations, but in general, we have about 30 genes that has this part EC number 1,14,13 and something else. So from 4,000 some genes, now we narrowed down to about 30 genes. And then we introduced this differential expression analysis. And based on differential expression analysis that has this EC number, we can eliminate all the genes that were not expressed in the presence of pyridine. So that will further narrow the list of gene candidates. Now what do I do? Still, there's a good number of genes, and not every gene get annotated with the proper EC number. So this is also the case here. Now I look for the genomic context. What I mean by genomic context is in bacteria, in prokaryotes, it's very likely that the genes are clustered together that act on the same pathway. And we see that all over most of the time in prokaryotes. So here I'm looking at the context of clustering data, and I'm right now picking the gene, the CDS3484, and I'm seeing that the whole operon that this gene is in is highly expressed. And for the gene 3483, right upstream to this highly expressed gene that has the partial EC number 1,14,13, has a EC number 1,2,1,3. That doesn't really help me. It's a very lousy EC number to, so to speak, because it's a very generic EC number, aldehyde dehydrogenase, it could be anything. So that doesn't give me any additional valuable information. This is where the PDB structural evidence comes really handy and really shines here to give us more evidence. So I run this app, the import PDB metadata into KBase genome that Chris has shown previously. And let me open this in a separate window. So this app give a wealth of information. What this app produces is the PDB structural hits that are homologous to Micrococcus luteus genes. And it gives the links to the PDB with the crystal structures that you can do your further analysis on the PDB site, and also give exactly what's the organism that the protein was crystallized, and also the core crystal structures that what the substrate are, and very importantly, also the literature that the study regarding the study. So now going back to this, my diagram here, I'm searching for additional evidence on this gene cluster, And I want to know what there's for CDS 3483, which doesn't give me any additional information from the genomics. And I'm searching is here CDS 3483, and I have 2 hits here. And so here it gives the bifunctional protein phenylacetate dehydrogenase, PAS-C, and that was crystallized in Escherichia coli homologous enzyme. And if I give and open this paper, and this explains the phenylacetate degradation pathway and the ring opening step, very, very key step. So phenylacetate is also chemically similar to pyridine, right? So, and we can hypothesize at this point, since it's very chemically similar, this 3483 can perform the same function on pyridine in a promiscuous manner. So this information is really important. That's why these tools are really shiny and very important to get additional evidence. In fact, we found out that the 3483 is in fact the enzymes, the ring opening enzyme in the pyridine degradation pathway, and we were that enzyme was able to characterize. So, and in addition to that, so we can also fetch the structures with this important PDB metadata into KBase genome tool that the QZ and going to extensively talk about these tools on her session. And here it's very I'm sorry, there is not so in this tool, the query RCSP database for protein structures, that QC is going to talk about this tool in her session, that you can simply paste a amino acid sequence. So I'm pasting here the Q for CDS 3483, that the gene of interest here. I can put multiple sequences. I can also combine SMILES or inch codes for compounds, and this will give me all the hits that comes from PDB. For crystallized structures, again, I can open, these are directly linked to the PDB site, and for further analysis. So in later in the session, in Brinda's session, she's going to talk about like query and learn from co-crystallized structures with the docking of the substrate for these potential gene candidates, and how you can align experimental and computational structures to aid binding site identification and functional characterization. So at this point, so I'm going to conclude my presentation, and I think we even have some few minutes if anybody have additional questions.
Thank you, Janneke. I'm looking at the chat now. Are there any questions for Janneke that you would like to, anybody in the audience would like to place in the chat?
I was so worried that probably I won't have enough time, but we do have some time.
I think you've done an excellent job, Janneke, and I'm sure that there'll be more questions as the afternoon progresses, and we'll be able to come back to you as needed, of course. So if you're finished, then it's my pleasure to introduce Dennis Peel from the RCSB Protein Data Bank. He's based here at Rutgers University in New Jersey. And the title of Dennis's presentation is Accessing Experimental Structures from the Protein Data Bank. Thank you, Dennis.
Thank you, Stephen. Hello, all. Can I just confirm people can see my screen?
You're good to go, Dennis.
Thank you. So good afternoon. I'm Dennis Peel. I'm a scientific software developer at the RCSB Protein Data Bank based at Rutgers University. Following from Janneke's presentation on the KBase workflow and how it makes use of PDB data, I'm now going to transition our focus specifically into the PDB archive and the structural data that's available there, as well as the tools that are available for accessing that data through the RCSB PDB. So I'll first begin by giving an introduction to what the PDB archive is, as well as who the RCSB PDB are. I'll then give an overview of our search tools, as well as some of the available download services that users may use. And I'll also give a discussion on the types of formats that structured data can come in. We'll then move into a live action tutorial in which I'll demonstrate some basic procedures on how to access data from rcsp.org. And this will focus using the case study of the Arabidopsis thaliana resistosome. So some things I'll cover are the basic searching and browsing of our data, looking at structure summary pages and exploring the data that's available there, how to download the data in various ways, as well as the programmatic manners of performing the searching and downloading tasks. And then at the end, I'll conclude with some additional resources and documentation that you may look at for reference on your own. So first, to provide a brief background, the Protein Data Bank, or PDB, was established in 1971 as the first open access digital data resource in biology, containing just 7 X-ray crystallographic structures of proteins. Since then, the PDB archive has grown to house more than 190,000 experimentally determined 3D structures of biological macromolecules, including both proteins and nucleic acids. These structures are determined experimentally by tens of thousands of researchers around the world using techniques such as X-ray crystallography, electron microscopy, nuclear magnetic resonance spectroscopy, and then they are submitted for deposition to the PDB archive, in which they undergo a thorough validation and biocuration process before they're officially integrated and made released to the public. This PDB core archive is jointly managed by the Worldwide Protein Data Bank, or the WWPDB, which is comprised of the RCSB PDB in the US, where I'm based, PDB-E in Europe, PDB-J in Japan, BMRB in the US and Japan, and EMDB in Europe. So while the RCSB PDB, PDB-E, and PDB-J provide access to the structural contents of the core archive through their respective websites, the BMRB and EMDB provide access to the NMR and electron microscopy data through their sites. We are committed to making sure that data adheres to FAIR principles, that is, that it remains findable, accessible, interoperable, and reusable, as well as ensuring that data is made freely available to the public with no limitations on usage. The RCSB PDB web portal at rcsb.org provides not only access to the contents of the PDB core archive, but also a rich collection of tools and features that you can use to search, browse, analyze, visualize, and download the contents of the PDB archive. Additionally, on a weekly basis, we furnish the collection of structures in the PDB with annotations from approximately 50 external data resources, such as UniProt, SCOP-E, CATH, and CIFS. And these provide additional biological and evolutionary context to the structural content. So we are always working to continuously add and develop new tools and features at rcsb.org to enhance the user experience and to give them more ways to explore our data. So for example, we've recently introduced a 1D, 3D protein feature view, which allows you to map residue-specific annotations from the 1D sequence directly onto the 3D structure, all through an interactive visualization tool. Additionally, this has been recently extended to groups alignment feature, which will allow you to align multiple proteins on top of each other. And both of these features will be demonstrated in subsequent talks by my colleagues, Brenda and Shuchi. So one major new development we've recently introduced that Stephen described earlier is the integration of approximately 1 million computed structure models from AlphaFold 2 and Rosetta Fold into rcsb.org, which we are now delivering alongside the collection of approximately 200,000 experimentally determined PDB structures. This integration tremendously expands the coverage of protein structure space encompassed at rcsb.org, filling in gaps in available structures for proteomes that were previously underrepresented by experimental data alone. And Brenda will discuss this further on. Last, alongside the data, features, and tools provided at rcsb.org, we also offer an extensive collection of educational and training resources to both aspiring and experienced structural biologists alike. And this is through the PDB 101 RCSB web portal. So when you navigate to rcsb.org, you have a couple different options for searching. The first is this top bar basic search. And this serves as sort of a Google-like type searching functionality. So you can enter in any set of terms, you can enter in a specific entry ID, or even an amino acid sequence. And when you hit the search button, you'll get results that are ordered in order of relevance. You can also fine-tune your search using the advanced search tool or advanced search query builder, where you can specify specific attributes or different qualities about the structure that you're searching for. And I'll give a brief overview of each of these in a second. The last thing I want to point out is you can also, if you're interested in programmatic usage, you can also click on this JSON button here once you perform a search, and this will generate the JSON query that you can submit to our search API, and you can integrate that with whatever programming tool you prefer. So to give a brief overview of each of the advanced search services that we offer, the first is the full text search. So this is, again, that full text Google-like search that I described, where it looks for structures that contain the terms that you search for and ranks them in order of relevance based on how frequent those terms appear. For a more fine-tuned control of your search, though, you can use these other search services. The first is structure attribute search, where you can search for structures based on a specific property about the structure. So you can look at all structures for a specific experimental method, like electron microscopy or X-ray crystallography. You can search for structures based on the author or the size of the protein. And we have a large variety of different attributes you can choose from. You can also search for structures that contain specific chemicals. So if you're looking for a structure that contains a particular ligand or cofactor, you can search for that using the chemical attribute search, or you can specify the name or the weight of the chemical. Sequence similarity search, as the name implies, you can look for structures that have sequences that are of a certain identity threshold to an input sequence. So you provide an input sequence and specify what cutoff you want for how close the sequence will be, and it'll return all the structures that have a similar sequence. Most of you have probably used a BLAST search at some point, but we use the MMSeq2 algorithm. You can also perform sequence motif searches, which is looking for a particular pattern of amino acids within a sequence. So let's say you're looking for all sequences that contain a serine, threonine, wildcard, glycine combination. Well, you can use this regular expression string in your search to find all sequences that contain that. The structure similarity search, as the name implies, you can look for structures that have a certain global structure similarity to the structure that you're querying. So you'd provide an input entry ID and chain ID to look for proteins that have protein chains that are similar global structure. You can also do this at a local level using the structure motif search. So if you're looking for a certain binding site, like the residues that comprise a particular binding site, you can search for that using the structure motif search. The chemical similarity search lets you look for structures that contain chemicals that are of similar structure to the input smiles or inchy strings, as well as you can even sketch your structure directly using the draw tool. You can choose your return type as well. So by default, structures are returned as entries. So this is the entire entry composed of one or more protein chains or nucleic acid chains. I shouldn't be strictly talking about proteins. You can also choose to return specifically polymer entities. So this would be either a unique protein chain or a unique nucleic acid sequence. You can also return your structures as assemblies, or you can get the non-polymer entities, and you can also get the molecular definitions. So molecular definitions refers to basically these chemical structures, the chemical definitions. And you can also consolidate your searches by grouping results. You can group them by various criteria. For example, if you want to, you can group them by sequence or of a certain sequence similarity. You can group them by deposit ID and as well as uniprot ID. So this allows you to more cleanly sift through your search results if you have a lot of them. So I mentioned earlier that if you were to click on that JSON button, you are brought to a search API query builder. And basically, this will just present you with the JSON object that you would use to search our search API service. And so you can customly build your query within our UI and then click on the JSON option in order to see how that query would look in your API call. And in fact, this is how the backend of KBase is working when it interacts with our website as well. It's using our APIs. So in addition to multiple search services, we also offer a couple of different ways of downloading data. So first of all, while you're browsing rcsb.org, you can download data directly from a structure summary page or an entry page. You can download all the data or all the structures from a search result page. So if you get a list of 10 different structures, you could download them right from that page. And you can also use our bulk download tool, which lets you enter in multiple IDs to download. Alternatively, you can also navigate our PDB archive directly or access it programmatically. So over HTTP, you can access our PDB archive at s3.rcsb.org, which is an Amazon S3 bucket of our PDB archive. It's also available at files.rcsb.org. And you can use any command line or scripting tools to access this, so curl, wget, or Python. And since the theme of this workshop is structured data, I wanted to emphasize that the structured data is located under this directory here. I'll give a quick demo of how to get there too, but this is where you can find a lot of the structured data. You can also use rsync and FTP. So when downloading data, when downloading structured data, it's important to be mindful in selecting what data format to use. Of particular note, it's important for us to emphasize that the current standard file format for data in the PDB archive is PDBX mmsif, which is data with the extension .sif or .sif.gz. And this is the official format, and users should use this. And if they're not using this already, begin to migrate to using this. For those who have used or worked with data from the PDB in the past, chances are you're familiar with the legacy PDB format file or with the .pdb extension. And I suspect many people may still be using this, but since 2014, we've stopped supporting extensions to this format in favor of the PDBX mmsif format. So in this new format, data is saved as a set of category and attribute pairs along with their corresponding values. And these categories and attributes are all defined in an authoritative dictionary, which you can find by browsing this site here. So this new format offers several benefits over the legacy PDB format. The first is that it supports structures that have more than 100,000 atoms, which is a limitation that the fixed column width restrictions of the PDB file format can't handle. The second major benefit is that it's extensible. And when I say that, I mean that when new experimental or computational methods arise that require their own type of metadata to define that type of experiment or to define that type of software, we need to add new categories and attributes in order to describe those new techniques. And so through this new format, we're able to do that because we can simply add new definitions to the dictionary to define those new types of metadata. So this is one of the main motivations for why we have transitioned here. So now as we begin to enter the tutorial part of the talk, let me begin by first introducing the case study that we'll be focusing on, and that is of the Arabidopsis thaliana resistosome. So Arabidopsis thaliana is the gold standard model organism in plant molecular biology. I'm sure that all of you at some point in your studies or career have come across Arabidopsis in your textbooks or your research. And the reason for that is because it's such a well-studied and extensively characterized organism. And like all organisms, it also maintains an immunity or immune system where it has to fend itself off of pathogens from the environment and maintain its survival. And one of the main ways that it does this is through the formation of the resistosome, which is shown here. So the way that this forms is through the sensing of a pathogen by a structure known as a nucleotide binding leucine-rich repeat receptor. And upon the sensing of these pathogens, these receptors oligomerize into a pentamer, which forms the fully active resistosome as illustrated. Ultimately, this process culminates in cell death, And the working hypothesis for how that happens is that this resistosome may embed itself into the plasma membrane of the cell and then create a pore and thus disrupting the plasma membrane. So the steps that it follows in forming the resistosome are illustrated in this schematic here. First, the NLR begins in its inactive state in which it's bound to ADP. Upon indirect pathogen sensing, it then undergoes a conformational change where it releases ADP, and then it undergoes another conformational change where it binds to ATP. Once it's bound to ATP, it then oligomerizes into the pentamer fully formed resistosome. And so for purposes of the tutorial, I thought it would be helpful if we try to actually find some of these structures, or at least the corresponding structures for these steps in the pathway, which have been published by Wang et al. And so when we do this, we can keep in mind some of these key elements of this pathway in developing our search. So for example, we can use the fact that this first inactive state is bound to ADP to help narrow down our search. We can also use the author and citation information. We can also use the fact that it's coming from Arabidopsis thaliana. We can also use the fact that these structures were determined by electron microscopy, which I found out by reading this paper here, and as you would be able to as well. So let's go ahead and try to do this. So I'm going to exit out of my presentation mode, and I'm going to go to rcsb.org. And so when we land on rcsb.org, we are presented with the top bar search. And so let's go ahead and just try to use that. So let's try to just search for Arabidopsis thaliana. And you'll notice that as I type, there's some suggestions that start populating in the drop down below. And if you were to click on any of these, it would allow you to perform a very specific attribute search for this specific keyword. But for purposes of the demo, I'd like to just perform a general full text search, and you can do that by hitting either enter, or you can press the search icon. And when you do that, you get approximately 2,400 structures. You can change the number of structures that are shown on each page by selecting from this drop down here. You can also change how these are sorted by default, they're sorted by relevancy score, but you can sort them by date, as well as other criteria. On the left, you have the refinements panel where you can see what type of structures are included in this result set. So these are some popular properties of structures that people like to refine upon. So they are included in the refinements panel. So although we search for Arabidopsis thaliana, we see some results from Homo sapiens, as well as other organisms. And that apparently must be because we performed a full text search in which these structures at some point in their metadata also include the words Arabidopsis or thaliana. So obviously we want to filter this down, and we want specifically Arabidopsis thaliana. So if we go ahead and check that and then click the refinements button, we've reduced our structure result set to about 1,800 results. And when we scroll up into the advanced search query builder, we can see that this property was added to the advanced search builder. So if you're not sure on what property to select, you can always select it from the refinements panel. But obviously we have to filter this down a little bit more. So let's try to add another attribute, and let's try to filter this down on the experimental technique that was used. So all we have to do is click add attribute. When I click that and I click this, you can see all the options that you have for structure attribute search. You can search by entry ID, deposit ID, structure title, author, release date, as well as methods, experimental method. And if you're not sure, and obviously this is quite extensive and it might be difficult for you to manually go through every single one of them, you can always just start typing it in. So you can just type experimental, and you'll see that experimental method would show up. So you can click that, and we know that the method was electron microscopy. And if we hit search, we've reduced our, oh, I'm sorry. I wanted this to be an AND operation. Hit search. There we go, voila. So now we reduce our result set all the way to 100 structures. So obviously though, 107 is still kind of a lot to go through manually. So let's try to add some more filters. The next one we can add is the author. So the author of the paper is Wang J. So let's add another attribute. And let's look for author. It's right there. And we can type in Wang J. So we hit search. Now we're down to 10 structures. It's great. So still 10, we can narrow this down even more because we're trying to find that one specific structure that has ADP bound. So how can we do that? We can use the chemical attribute search. So let's open up that part of the advanced search builder. And we can select chemical IDs under chemical components. And you see it even provides some example queries of what you can use for values. This is to help users try to figure out exactly how to format or what type of values that the property is looking for. So we can type in ADP. And if we do this, we're down to 2 structures. And so now from these 2 structures alone, we can quickly tell which one's the right one. And the way we do that is we can simply use the citation information. So this is the 2019 paper from Wang J et al. So we have successfully found our structure. And when we click on that, we're brought to the structure summary page, which contains some general information about the structure, as the name implies. It provides the title of the structure, some information about where it came from, when it was deposited, experimental data, validation information. You can also download the structure directly from the page. So if you were to click on this dropdown, you can download the FASTA sequence, the PDBX MMSIF data file or the compressed file, or the PDB format file. But again, please try to begin migrating to the MMSIF as this will continue to be supported moving forward. You can download other related information. We also link out to the corresponding experimental data at EMDB. And if you scroll down further, we have the citation information, and then we're provided with a breakdown of the structural contents of the entry. So as every entry contains at least one or more protein chains, this is a breakdown of each of those unique chains. So an entity is a unique protein chain. And so the first entity is RKS1, and the second entity is disease resistance RPP13 like protein 4. So this contains 2 unique chains. And if we scroll down further, we see what small molecules are included here. And as we search for it, it contains ADP. And then we have more information about the experimental and validation, as well as the deposition history and revision history. If we scroll back up, we'll see that there's a number of different tabs up here on the structure summary page. Each one brings you to a different aspect of the structure. So 3D view would bring you to a 3D view of the structure where you can actually interact with it, play around with it, click on it. And this will be demoed more thoroughly by Shuchi later on. So I'm going to go ahead and go to annotations. And this illustrates some of the externally integrated annotations that we provide for our structures. So this provides annotations at an entity or protein chain level. And as you see, we have some ECOD annotations, protein family annotations, gene product annotations. And then we can also go to experiments, where we're provided with information about the experimental conditions used to solve the structure, to determine the structure. And if you go to sequence, you get an overview of residue-specific annotations. So you can see the level of disorder for different residues. And sorry, I kind of did that without explaining. You can zoom in by scrolling. So if you zoom in, you can see the specific residues. And if you hover over any of these in the upper right, you see the information about that annotation. And then you can even select what chain you want to look at. So if I selected chain B, we see we have most of the same annotations, but in addition, we now have this additional row for binding site of ADP. So this signals to us that the entity that we were looking for is entity 2, because this is the one that binds to ADP. This is the NLR. When you, just to complete this story, when you go to the genome tab, we have genomic information about the entry. And we also provide version information about the entry. So like when any changes are made. So now that we know that the corresponding entry or entity is the second entity of this structure, that is the one that's bound to ADP, we can scroll down here and we can see that we're provided some options to link out to UniProt searches. So by clicking on this link here, you could search for all structures in the protein data bank that have this UniProt ID. And this will be what we want to do because we know that in this pathway, that NLR never actually changes its sequence. The sequence remains the same. The only thing that changes is its structure and what it's bound to. So we can use that as a way to find the remaining structures in that pathway. So if we click on this, and I'm going to open up a new tab, we see that we're presented with 5 structures. And scrolling through them, we see that they're all from the same literature citation and from the same authors. So this represents the complete set of structures that we originally set out to look for. And now that we have these, if you're interested in downloading them, you can choose to either click on each one individually and download their structure files from there. You can download the structure files by clicking on this button. But even more efficiently, you can click on this button here, which will download all selected files. And if you click on that, it'll bring you to our batch download tool, which lets you specify multiple structures to download the structure. And here you would just select PDBX MMSIF as your data format, and then you would generate the file for batch download. I'm going to go back because I'd like to also describe some of the other links that exist on the structure summary page that I kind of skipped over. You'll see these plant protein or Arabidopsis thaliana. Any of the links that you see on the page will, most of them will let you perform a search for that particular structure attribute. So if you're on this page and you wanted to then look at all the structures for Arabidopsis thaliana, you could click on this as the organism. So that's another helpful feature about our summary pages is the ability to link out and perform different types of similarity searches. You can search for structures by similar 3D structure or sequence. So we offer a variety of options for facilitating searches throughout our site. So I wanted to go back though to this search page that I had. All right, so we kind of developed this rather complex query. How would we do this through programmatically? Well, the simplest way to do that would be to click on this JSON button as I described. If I click on this, it brings us to the RCSB PDB Search API Query Editor. And then it automatically populates that editor with that corresponding JSON for the query. And if you press this play button here, you get the results that we just had on that page. You get a list of the identifiers along with the corresponding relevancy score for those structures. By default, these searches are restricted to the first 25 results. So if you want to get more results, you can either increase the row number or you can continue to paginate, which is the more programmatically friendly way of querying an API. So along with this programmatic search query option, I also wanted to just quickly still illustrate how you can access our PDB archive directly to download files. So you can go to our documentation page and go to programmatic access. And here we provide a lot of documentation about how to download our data. And one of the things I described earlier was that we now offer our data on an AWS S3 bucket, which you can access here. So if we open this up, this will bring us to the PDB archive. And if you're looking for a specific structure, and let's say we're looking for the one that was on our search results page. Let's say we're looking for 6J5W. We would go to under public, PDB, data, structures, divided, MMSIF. And obviously you can see we offer all the formats on here, and we offer a lot of different data like NMR data and so forth. But for purposes of the tutorial, I'm just kind of quickly navigating through to find the specific structure. And again, this is being recorded, so I hope that you'll be able to review it later in case you miss anything. So we're looking for 6J5W. So to get there, we need to use the 2-letter hash, and that's going to be the middle 2 characters. So it's going to be J5. And so we can even just search for it here. If we type J5, and then we were looking for 6J5W, 6J5W. And voila, we have our structure. So we can click on that. It'll download the structure. And now you've been able to use our PDB archive directly. And again, you can use your own curl or Python scripts to perform these downloads as well, which is probably the more useful way of using our PDB archive directly as opposed to navigating manually. But you can always navigate it manually if you wish. With that, I just wanted to provide a quick summary of what we did. I illustrated some of the interactive methods that you can use for searching data and downloading data, as well as some programmatic methods you can use for searching our data and downloading our data. And then I wanted to provide some important links that you might find helpful when you want to try to follow along with any of the things I demonstrated. And with that, I'd like to thank you for your attention, and I'd be happy to take questions.
Thank you very much, Dennis. Are there questions for Dennis in the chat? I'm not seeing any. Several queues are in the doc and being addressed here.
No, there were 2 questions in the Google Doc.
Oh,
we already
okay.
addressed. So
All right. Oh, okay. Terrific. Thank you. So we're keeping it running.
people are
running.
paying attention and they are asking questions.
Okay. I'm just looking in all the wrong places. Okay. Well, that says something about my life, doesn't it? So, are there any questions for Dennis from the KBase team? Okay, well, hearing none, it's a pleasure for me to introduce Brenda Vallat, another member of the RCSB Protein Data Bank team here at Rutgers University. And the title of Brenda's presentation is the flip side, accessing computed structure models generated using AlphaFold 2 or Rosetta Fold. Brenda, the floor is yours. Thank you.
Okay. Thank you, Stephen. Sorry, I have to get my PowerPoint. Okay. I just want to make sure everyone is able to see my slides.
You're good to go, Brenda.
Okay,
Yep.
thank you. Thank you very much. Thank you for the introduction, Stephen. I'm Brenda Vallat. I'm a scientific software developer at RCSB Protein Data Bank. And today I'm going to be talking about finding, accessing, and analyzing computer structure models from rcsb.org. All right. I have a brief outline here, but I realized Stephen already introduced what computer structure models are, so I will kind of skip through that quickly and move on to explaining the models that are currently available in rcsb.org, and then show a live demo to illustrate the tools and functionalities that we have on rcsb.org. And I will focus on case studies that have already been introduced by Chris, Janaka, and Dennis. The disease-resistant RPP13-like protein from Arabidopsis, the riboflavin riboflavin biosynthesis pathway from Arabidopsis and the Micrococcus luteus aldehyde dehydrogenase. What are computed structure models? We already saw part of this slide in Stephen's talk. These are, so I won't spend too much time, just to quickly catch up. These are predicted models of macromolecules obtained using computational methods. And the two recent methods, AlphaFold 2 and Rosetta Fold 2, are ab initio prediction methods. AlphaFold 2 is developed by Google DeepMind, and Rosetta Fold developed by Howard Hughes medical investigator David Baker and his team at University of Washington. These methods achieve accuracy comparable to low-resolution experimental structures, and their success was enabled by recent breakthroughs in applying artificial intelligence and machine learning approaches to structure prediction. These methods have been trained using thousands of sequences and structures of known proteins from the PDB. So the primary data for training these methods is coming from the PDB. So without the enormous wealth of curated and validated 3D structure data from the PDB, the application of machine learning techniques to train these systems would not have been possible. AlphaFold Protein Structure Database provides open access to AlphaFold 2 predictions. The database is hosted at the European Bioinformatics Institute and includes over 200 million protein structure predictions, providing a broad structural coverage of sequences in UniProt. The Model Archive is a repository for computed structure models referenced in publications and is hosted at the Swiss Institute of Bioinformatics and includes predictions of core eukaryotic protein complexes modeled using a combination of Rosetta Fold and AlphaFolds generated by David Baker and his team at the University of Washington. So these two are the primary resources for the computer structure models, which have now been integrated into rcsb.org. I want to quickly touch upon model quality metrics when it comes to computer structure models. Although these computer structure models generated with the recent machine learning approaches have become increasingly accurate, it is important to look at the model quality estimates for these predictions, because these can be, the estimates can be used to evaluate these models and assess their suitability for specific downstream applications. Computer structure models in AlphaFold DB and Model Archive include two types of model quality metrics. The first one is a per residue confidence measure called the Predicted Local Distance Difference Test, PLDDT, provided on a scale of 0 to 100, 100 being the best. As can be seen here, regions of high and low confidence based on PLDDT are identified using color codes. The blue and cyan regions are the regions of high confidence, and the orange and yellow regions are regions of low confidence. The PLDDT score can be averaged over all residues in a protein chain to provide a global score. The second metric is the predicted aligned error, or the PAE score, which is the expected position error at residue X when the predicted and true structures are aligned on residue Y. PAE enables the assessment of residue contacts as well as domain orientations. So these are the two kinds of quality assessment metrics that are available from AlphaFold DB and Model Archive. Before delving into the computer structure models available in rcsb.org, I want to give a brief introduction to the Model CIF data standard. Dennis mentioned the PDBX MMCIF data standard used by the PDB archive. Model CIF is an extension of PDBX MMCIF for computer structure models. Definitions for representing small molecules and macromolecules and atomic coordinates are all taken from PDBX MMCIF. And definitions pertaining specifically to computer structure models, including definitions of targets, templates, alignments, coevolution data, model quality metrics, all these are defined in Model CIF. Model CIF is publicly available via GitHub and is managed by the Worldwide Protein Data Bank Model CIF Working Group. Importantly, The ModelSIF data standard is supported by both AlphaFold DB and Model Archive. Moving on to computer structure models in rcsb.org. This summer, we released an updated rcsb.org research-focused web portal, which now delivers over 1 million computer structure models, together with the approximately 200,000 experimental structures already available in the PDB, thus creating a one-stop shop for studying macromolecular 3D structure data. This was made possible because a ModelSIF, which is an extension of PDBX MMSIF, facilitates interoperation with PDB data. The extensive set of tools and services provided by rcsb.org can be used to search, analyze, and visualize computer structure models alongside experimentally determined PDB structures. These computer structure models in rcsb.org are consistently distinguished from the experimental structures through source-specific icons and unique coloring schemes. Experimental structures are identified using the blue Erlenmeyer flask icons, as you can see here, and the computer structure models are identified using the cyan computer monitor icons shown here. Provenance information about these models are clearly provided, and we have extensive documentation that explains how to access and analyze these models alongside the experimental structures. So what are the models that are currently available that have been integrated with rcsb.org? The current set consists of a prepackaged collection of about 1 million models from AlphaFold DB, as well as 1,100 models from Model Archive, comprising of the core eukaryotic protein complexes modeled using Rosetta Fold and AlphaFold 2. These computer structure models collectively cover the entire human proteome and those of model organisms, selected pathogens, as well as organisms relevant to bioenergy research. I will now move on to the tutorial, the live demo part of this session, and showcase a few case studies that have been introduced by Dennis, Janaka, and Chris earlier today, including the Arabidopsis thaliana resistosome, the riboflavin biosynthesis pathway, and the aldehyde dehydrogenase from Micrococcus luteus. My goal here is to use these case studies to demonstrate the tools and functionalities that we have on rcsb.org to study these computer structure models. So now I will be moving on to my browser. Okay. So let's start with the first example, the disease resistance protein from Arabidopsis thaliana. I just want to copy the term that we want to search. We start with the top bar search. The first thing that I want to point out is including computer structure models is an opt-in button here. You got to turn that on if that needs to be included in the search. We'll do that right now. And then I'm going to paste the search term. Auto suggestions come up here, and it's always recommended to use these if they are relevant to your search. So let's start with the UniProt molecule name for this specific protein. And this launches the search query that returns 29 structures. A quick look at the refinements panel shows that there are 24 computational and 5 experimental structures. The refinement panel has terms that are, now that we've included computer structure models, there are some specific attributes that are relevant to computer structure models. You can filter based on the source database or the global quality metric PLDDT score. Or you could look at just the computational or experimental structures. In addition, the search results can be sorted based on either the global quality metric So then you can see the best scoring ones on top, or you can sort based on, okay, you have a combination of experimental and computational structures, but say I want to look at experimental structures first, you can do that, and the results would be sorted accordingly. As I mentioned earlier, the experimental structures and computed models are distinguished clearly using color schemes and icons. You can see that here. And if you scroll back up, you can see that the advanced search query is populated with the query term that we actually started the search with. And let's say we want to now look specifically at Arabidopsis, we can filter for Arabidopsis. And now there are only 6 structures. This includes the 5 from experimental and 1 computational structure, computational model. The advanced search query builder also has specific attributes for computer structure models. You could search based on the search source database, global quality scores, or identifiers from the source database, either from AlphaFold or AlphaFold DB or Model Archive. All these can be used to build into the query builder, and complex queries can be built using other scientific search queries like sequence similarity or structure similarity, all these that are useful even for experimental or if you're searching for experimental structures as well, like Dennis already pointed out. Now let's look at a computer structure model, the structure summary page, complementary to the experimental structure summary page. It has a little less information than experimental structures that Dennis demonstrated a little earlier, but I would like to point out a few specific attributes that are highlighted here. One is the model confidence, the global confidence score, and the distribution of local confidence score based on the color scheme shown here. The image also follows the same confidence PLDDT-based confidence color scheme. And you have a similar macromolecule section to what you see for experimental structures, that you can launch different kinds of queries from this page, and also the 1D protein feature view. All right. Now, I would like to move on to the next case study, which is the Micrococcus luteus aldehyde dehydrogenase. So in this case, I want to start with the sequence. So I'm going to the advanced search query builder. Sorry, I want to launch the sequence similarity search. I have the sequence of this protein. Let's use all the default parameters, but I want to return polymeric entities, and I want to include computed structure models. And if you launch that search, that retrieves 666 entities here. But what's of interest is that the highest scoring one has a sequence identity of 48%, which means there is no 100% match. So this exact sequence, there is no structure currently available either in the experimental PDB archive or the computer structure models that have been integrated. So at this point, you could either use one of these experimental structures as templates to build a homology model, or you could use AlphaFold software to build an ab initio model. So using the KBase pipeline, we have an AlphaFold model built for this structure. And the next, the interesting question here now is, okay, I have a model, what else can I do? The sequence search has given me a lot of interesting results. Let's say we pick an example here. And this happens to be the structure of the bifunctional enzyme from E. coli. And this, this structure has small molecules bound, crotonal coenzyme A and NADP. So what we can do is, okay, this turned out to be structurally, I mean, in the sequence search, this is a close homolog. So we could do a pairwise alignment. We have a pairwise alignment tool on rcsb.org. So let's launch that. And okay, so the E. coli enzyme is 6JQO. So the tool allows you to either choose an entry that's already in the database or upload a structure that you might have. So let's choose this one, 6JQO. And I have I'm going to choose upload file here. And I have the Micrococcus luteus aldehyde dehydrogenase that was modeled using AlphaFold that was shared with me by QZ. And the pairwise alignment tool has several options for the alignment, but for now, for this demo, let's just choose the default option and hit compare. This is going to, this takes a little bit of time, so, okay. So we have the E. coli enzyme and the uploaded file that has the M. luteus alpha-fold aldehyde dehydrogenase model. And if you scroll down, our MolStar visualization tool has now shows the aligned structures, and you can visually see that they are very similar. In addition, you could click on the scores tab and you can see that the RMSD is 1.31. There's a TM score 0.93, which is very significant. So these are structurally similar. By default, the view it shows the aligned residues. But if you use the pull down menu here and look at structures, what it does is the E. coli enzyme is now superposed, that the entire assembly is superposed to the Micrococcus luteus alpha-fold model. And now there are ligands, small molecules present in the E. coli enzyme. We already saw that. So now what you could do is you can focus on these ligands using the MolStar tool. Nishuchi will explain in more detail about how to use the tool, but I just want to quickly highlight how you can get some structural and functional insights using the tools in rcsb.org. So now you can focus on the ligands, and so there are ligands in the aligned regions here, the crotonal coenzyme A and NADP. So this you can focus and you can get some additional insights into the ligand binding and other functional insights into the aldehyde dehydrogenase and what kind of ligands can bind. So this kind of information that experimental structures have these small molecules bound and the computational models don't have that. So doing this kind of a pairwise alignment can give additional insights into small molecule binding. All right, so the next story that I want to point out is the pyridine binding, the pyridine degradation pathway for the same enzyme. We want to see if we can find structures that have pyridine bound. So let's start with pyridine again. I'm going to use the auto suggest. Scroll down to in chemical name pyridine. Okay, let's turn CSM on. So that pulls up several structures, but it could be a derivative of pyridine as well. We want specifically pyridine, and that happens to be this ligand 0PY. Let me just show you the ligand page. This is the ligand page for pyridine, and it says it's present as a standalone ligand in 3 entries. And these are the 3 entries that have pyridine. So for example, let's click on the first one and go to the small molecule section. There's pyridine here. A cool feature here is if you click on this ligand interaction button here, that takes you to the 3D view directly, but it focuses on the ligand and the interacting residues. So you can already see the pyridine ligand and the residues in the protein that are interacting. And what we can do from here is we could do like a structure motif search. We can launch a structure motif search, but what that means is you can select some of the residues that are interacting with the ligand. Like for example, I can choose some of these residues. As I'm choosing, you can see that the query, you can see what is being chosen here, and then you can launch a search right from this the Molstar viewer here. I already did this search, so I want to, yeah, okay. So I had picked randomly a few residues from the ligand binding motif, and then launched the structure motif search, and that brings up 481 results, and all of these have this motif, but in this case I just randomly picked a few. You could look at closely which are the residues that are interacting with pyridine and search for that particular motif. And this now has a included computed model. So now you can see there are computed structures as well as experimental structures that have this motif, and that can you can look at the results in detail and see which of these computational structures that have this pyridine motif, and if there's any overlap with the sequence search that we started with, to compare the results of the sequence search with this struct motif search to see if any of the structures of interest have this particular motif. So this also can give additional functional insights into ligand binding in computed structure models. Okay, so the last story I want to focus on, let's go back to, is the riboflavin biosynthesis pathway. So we saw that we have the E. coli enzyme, which was, I think from Chris's story, it was 207P. Let's make sure that's what it is. Let's see, 207P. Okay, so that's the E. coli enzyme. So I want to, and then we have the 207P chain A, and now we have the Arabidopsis enzymes, the deaminase, and the reductase. So I want to pick one. I have the identifiers here, so I just want to, okay, chain A, and then I can launch a pairwise alignment again. Okay, so this is the E. coli rib-D with the Arabidopsis, the deaminase AlphaFold model. So now we can do, again, a structure comparison, pairwise alignment, to see how these structures match. The scores look good, and then you do the same thing, look at structures so that you can highlight the ligands and see where the ligands can bind in the computed model. You could do the same thing with the reductase. Let me go and get the accession code for that. All right. Right. And then I could do a comparison Okay, so that's the E. coli ribd with the Arabidopsis reductase. This one is not as good a match as the deaminase, but still it's reasonable. A TM score higher than 0.5 essentially means a similar fold. And you look at the aligned residues. Yep, looks reasonable. And then you can look at the entire structure. And now you have the context to look at ligand binding and delve into the details of, okay, where can ligands bind, small molecules bind, and what kind of functional insights you can get. Okay. So one other thing that I wanted to point out before closing is you can also do a structure similarity search. This is another tool that you can launch from an entry page. So if you click on that, and I always recommend to go and look at the advanced search query builder when you're clicking on any of these query by example to know what search is being carried out, and you can always build on top of this, add subqueries. And let's turn this on and search. So in this case, it's doing a shape search or a structure similarity search and coming up with both experimental and computational structures that share structure similarity to our query here. In this case, I think I ended up using the Arabidopsis bifunctional, no, not the, sorry, the E. coli bifunctional enzyme. And based on the results that you get, again, there is a structure match 3D view button here. And when you click that, it'll take you directly to the pairwise alignment and align the query to the result that we picked. And you can see the structure similarity again. All right, so we looked at different tools, the pairwise alignment tool, the motif search, sequence search, structure similarity search, the different kinds of scientific searches that you can carry out using the rcsb.org web portal. And this brings up, I mean, you can include computer structure models or focus on experimental structures depending upon the scientific question. So to quickly summarize, rcsb.org now provides access to over 1 million computer structure models from AlphaFold 2 and Rosetta Fold alongside approximately 200,000 experimental structures of macromolecules. Various search, analysis, and visualization tools available on rcsb.org facilitate the study of these models together with the experimental structures to obtain additional structure and functional insights into the binding sites, etc. And depending upon the scientific question being investigated and the data available to address this question, for example, sometimes you might only have the sequence, or sometimes you might have already an experimental structure, or sometimes you only have you have good templates from which you can build a model. So based on the question that's being investigated and the data that's available to address the question, different search and analysis strategies can be devised using the tools available on rcsb.org. Thank you. I can answer any questions.
Thank you very much, Brenda. I'm just looking in the Q&A. And there's a question from Yujun. How can you know whether the alignment of the protein is good or not?
If you're talking about structure alignment, as I told you, you could look at the scores here. The root mean square deviation is one score that tells you how close the aligned structures are in terms of the atomic positions. And also the TM score is another structural alignment score that gives you a good idea about whether an alignment is good or not. So closer to 1 is always good. closer to 0 is not. And 0.5 or above means it's at least the same fold. So the TM score and RMSD can be used to get an idea about how good the alignment is. And if you're looking for sequence alignment, let me see if I have a tab that has, no, that's the structure motive search. I wanted to. So if you're looking at sequence alignment, there will be the sequence alignment score. There's the E-value as well as sequence identity. Those are the scores that come up when you look at sequence alignments. And those scores can be used to see whether the alignment is good or not. So as I mentioned, anything about 40% identity tells you this is a very good template based on which a model can be built for your query.
And Shuchi, it looks like you wanted to jump in. Please feel free to do so.
No, I didn't want to jump in. I was just saying that the answer was already being given live.
Okay, understood. Yep. Okay, terrific.
And the
Thank you
same
very
is
much.
also, I think, in the document, and I answered it in the document too.
Thank you.
Oh, okay. Thank you, Shuchi.
That's terrific. It's excellent,
I'll let Brenda
excellent
proceed.
teamwork.
I wanted to see her answer too.
Good. Well, I want to thank our first 4 speakers for sticking to time. It's we're coming up on the break with just a few minutes, just actually 4 minutes ahead of schedule. So we're going to reconvene promptly at 3:15 p.m. Eastern time. And when we'll hear from Shuchi Dutta, and she'll be talking about actual visualization of PDB structures and computed structure models. So let's take a quick break, stretch your legs, and get a bit of fresh air perhaps, and we'll see you in now 18 minutes. Thank you. It's my pleasure to introduce Shuchismita Dutta from the RCSB Protein Data Bank team here at Rutgers. And she's going to be introducing the MolStar molecular graphics system, which is our workhorse for 3D visualization. Over to you, Shuchi.
Thank you, Stephen. I am going to share my screen. And I hope that you can hear me and see the screen.
I can hear you. I'm just waiting for you to go into presenter mode.
So I'll actually stick with this mode because I'm going to go back and forth between.
Understood. You are good to go.
Okay. So good afternoon, everyone, and welcome back after the break. So in the first part of the course, we heard about various ways to calculate models and find structures and models in the rcsb.org or from other data resources. What we're going to focus on in this session is visualizing that structure or structures that you have identified or computed. Now, this is one of the most fundamental things that you probably want to do with the structure is to see overall what the shape of the molecule is, and then also examine closely specific regions of that particular structure, perhaps a particular residue, how a ligand is binding, where the interaction faces between your protein of interest and its partner protein or nucleic acid. You want to also examine and understand how mutations may impact the structure and interactions of a protein. So all this, to do that in a visual way, the tool that we're going to use here is MolStar. And I'm going to present MolStar as a tool to examine one particular structure. That's where we will begin. But then I will also show you a standalone MolStar, and the relevance or importance of that will become hopefully evident when I get to that point. And then finally, I'll also show how the MolStar tool has been woven into various tools and functionalities that the rcsb.org website offers, so that you can not only look at the structure, but also map on it various annotations. You can look at trends and patterns and things like that, that might give you insights into the function and biology of your protein or molecules of interest. So these are the 3 things that I plan to cover in the session here. And let's get started with the first part where I'm going to talk about exploring one particular 3D structure. And in this case, I'm going to start from the structure summary page that was introduced to you in the previous sessions. And to do that, I'm going to go back and forth between the slide deck here and the website actually, so that you can, if you are in front of a browser where you can do this, you're welcome to follow along. So this is the rcsb.org homepage, and let's just start with a simple example. 4-HHP, this is hemoglobin. This is not an Arabidopsis or M. luteus or any other such protein. We'll come back to an Arabidopsis example later, but this is a quick way for me to introduce you to the tool. So how do you access this MolStar tool? So on any structure summary page, whether it is an experimental structure or a computed structure model, the 3D view tab, this will open up the structure, or at the bottom of the page, at the bottom of this little graphic, there is this link called structure that will also take you to that same page. And I wanted to spend a couple of minutes getting you oriented to this. So this is the MolStar interface, and the white space here where you see this colorful molecule, you can use your mouse to rotate things, whatever is being displayed here. This is our 3D canvas. On the top up here is our sequence panel. So notice for every polymer that is displayed in this, the sequence of that is shown up here. And if you click on something there, the same is identified and clicked here. So if I click on something here, notice in the sequence panel, it gets highlighted. So there is a connection between these two. This panel on the right is the controls panel. The controls panel allows you to do many different things. The top part is telling you something about the structure. So are you looking at the assembly? This is the default, but you can also look at the symmetry mates. Since this is a crystal structure, it computes the symmetry mates. You can go back to the assembly. The measurements and other panels we will come to in a little bit. The structure motif panel was already demonstrated by Brinda when she showed how you can select specific residues and do this. But let's start here with this components. The components is actually showing you what is being displayed here. So there are polymers. If I hover over this, it is highlighting all the polymers. I can hide that or show that. There are ligands. So in this case, this is a hemoglobin molecule. So the heme ligands are here. So I can hide that and show that. Waters, the same thing. And an ion. So there is I think a phosphate ion in this structure. So that's a separate component. Now, this is the default display. I can create any combination of ligand and residue or different chains and create components out of them. And I'll show that in a moment. But those can be added to this list and they can be independently turned on and hidden and things like that. If the particular structure has any experimental data, such as electron density or EM map, you can turn that on here. The quality assessment, if you remember, Brinda talked about the PLDDT scores. So PLDDT for computed structure models and the EM sorry, not for EM, for X-ray structures, there is a quality assessment that is available for display here, which not in this, but I think I can show it to you in 2D and 2. That's another structure. This is another hemoglobin structure. And in here, if I look at the quality assessment, and so the blue regions are the higher quality and the orange and yellow regions are the lower or less ordered regions. And this is being designed to mimic or be similar to that PLDDT coloring scheme that you had seen earlier. So let's go back to our 4-HHP. And okay, further down here, you have options to export animations. You can export the model and do a bunch of other things. So all these functionalities are covered in the controls panel. And again, this is your 3D canvas and sequence panel. So back to our So these were some of the key features that I wanted to point out. We've already covered all of them. So let's go on further. Now, when you upload a structure into your tool, perhaps the first thing you want to see is what does this molecule look like overall? Now, the molecule may be a single protein chain or a complex of multiple protein chains with nucleic acids and carbohydrates and whatnot. So you want to see the whole thing first and see what all is going on in here, and then perhaps zoom in and try to understand what is unique to what is happening around a ligand or a particular active site or whatnot. And so the MoldStar tool functions in two different modes. And so let's go to our demo session. So here, if I, in the default mode, if I move this around and click on, so I'm clicking on this heme, you can see it's showing up as heme 142 at the bottom. If I click on this, it'll jump in, center around that particular heme, and show all the amino acid side chains within five angstroms of that particular ligand. It'll also show you through these dashed lines the types of interactions that are stabilizing or holding that ligand in place, right? And so this is very easy and a quick way to understand what is going on in the neighborhood of your ligand of interest or a particular residue. I can go to any residue and say, I want to see what is going on in the neighborhood of this residue, okay? So you can go to any residue. So this is the default mode. But so this is great if you want to pick a particular residue and say, okay, show me what's going on around here. But sometimes you may want to select a number of residues. So what do you do then? You can't keep jumping around. And so for that, you activate the selection panel by clicking on this toggle menu, this little arrow. Notice here, a new menu has now shown up. And with this new menu turned on, now I can either click on a number of residues in here and see those are getting selected in here, or I can click on anything on the 3D panel. And if I click on a white space, all the selection is gone. So this is a quick way for selecting things. And then I can say, I want to do something with this. So why would you want to select? You've selected and you may say that I want to color it in a different color, or I want to see it in a different representation. So for example, let's look at this particular heme. So I'm going to select this heme. And I want to display this in a space fill representation because I want to see how much volume does it really occupy in this cavity that it is sitting in. So to do that, I can click on this and say, change the current selections representation to space fill. And I'm going to give it a name. So I'm going to call it heme1 and create component. So voila, now my new component, notice a new component was introduced here, which is showing this heme group in a space fill representation. The components do not have to be a ligand. They do not have to be a polymer chain. They can be any combination of chains, residues, whatever it is that you want to look at in one go. And I can hide this and show this. And depending on what it is I want to do with this, so perhaps I'm trying to create a publication quality figure, I may want to change the color of this and do all kinds of other things. To change colors, I can click on this and say, I want to color the whole thing yellow. Okay, things like that. And I can hide that. Okay, so we learned how to quickly look at the neighborhood of a particular selection. And also we learned how to select different things. And so these two, focus and select, these are two things that we are able to do. And we learned about that. Now let's go to the next thing. We also actually quickly looked at how to color things, how to create new components, and how to select things. So the selection, if we go back to our thing, I can say that in this, I want to select everything that is a ligand. So it's asking me which ones do I want to select, and I can say I want to select in one go, I want to select all the heme groups. So it's picked up all that. Okay, And similarly, there are other ways of selection that you can use using the sequence panel, where I can go in and say, okay, I want to select all the hemes. And so now I unselected it. So there are several ways to select things and several things that you can do once you have selected your residue or chain of interest. The selection level, as you may notice, right now we were in the residue level. We can select chains, entities, or go, more granular. So we can even select at the atom level, and we'll look at that in a moment. Okay, so we've learned how to display color. Measurements. Now, one other thing that you may want to do when you're visualizing your structure, you may ask questions like, how far away is this atom from that atom, or this ligand from that ligand, or this chain from that chain, and so on. So for example, let's go back to the beginning and say, so we know in the case of hemoglobin, there is this proximal and distal histidine, which is sort of holding that heme group in space. And I wanted to find, or let's ask this question, how close is this heme group to this proximal histidine? And so the easier way to do this would be I can just jump in here, click on this, jump in here, and zoom in to focus in around the heme. Now, the distance that I want to measure is this distance. Okay, so I'm going to activate the selection mode. And instead of being at the residue level, I'll go to the atom level. So now when I click on this atom and that atom, I have selected just the atoms, not the whole residue. And in the controls panel, if you open up the measurement, you will see that these two selected atoms are already showing up. In order to label, I need to select only one atom. That's what's listed here. For a distance, I need 2 atoms, and I've already selected the 2 atoms. And if I click on distance, it is showing up as 2.14 and 2.14 over here. And if I wanted to do more than that, if I wanted to measure an angle, I would have to select 3 atoms, and for dihedrals, 4 atoms. And so this is another quick way of doing some analysis to see what your structure is telling you about the function or the biology of that protein. It is possible that under different conditions, in the presence of different ligands, cofactors, environmental conditions, etc., that distance may change. Those bonds may become, longer, shorter, whatever. So here's a quick tool you can use to measure and compare things. Okay, so now what I'm going to do is all the things that I was showing so far, they were directly interacting with the structure. So you open the structure in this 3D canvas, and you were trying to examine the neighborhood, measure distances, change representation, so on and so forth. But can I use this tool to learn more about what is known in the scientific literature or from other data resources about this particular protein or assembly? And so to do that, let's go back to the structure summary page and notice this 1D, 3D view. This will actually, if I click on this, this will open a view where on one side, the sequence and 1D annotations are provided. On the other side, there is this 3D structure. And these two are connected so that if I rotate this and click on something here, it gets highlighted here. So what can I do with this? This top part is information derived from the structure. Things like, outliers and binding sites. So where are the heme ligands binding? Where is this? So right now I'm looking at chain A, where along the chain A are the chains B, C, D interacting. So those are highlighted here. But in addition to that, information from structures, if I scroll down, I can actually see things, annotations that are being brought in from PFAM, CATH, and various other resources. So for example, here is a glycosylation site. And again, I can zoom in to look at the residues. And so this is a lysine that is glycosylated. So if I click on this, in the 3D space, it'll actually jump into that lysine and show me. So this particular structure does not have the glycosylation, but I can quickly examine that where is this residue located in the overall structure, and what would happen if this was glycosylated. So here, right now, there is some other residue and thing, and so I don't want to get into the details, but I can turn this off. And using this left-hand panel where the various annotations are, I can look at what is going on or where are those particular residues or interactions located in the structure. Okay, so this is helping us gather information from other resources onto the structure, map it, and then maybe that'll help us ask new questions or develop hypotheses, etc. So again, as a quick summary, on the left-hand side, there was the sequence panel, the 1D panel, this is the 3D panel, and they are connected in a manner that if you click on either, you can select residues on both. On the top part, we saw structure features. On the bottom part, we saw various annotations from the various data resources that we connect with and integrate information from. So this is all that I wanted to talk about in terms of the examining one particular structure, right? And although the ligands, etc., will not be available for CSM, the computed structure models, because they don't have them right now, but other annotations and other things that you can do, the same functionality of 1D, 3D connectivity is available for those structures too. Now, the next thing that I want to point out or show is a standalone MolStar tool. Now, if you haven't already figured out, what I started with was from the structure summary page, but what if there is no, your model is not in the PDB, or it's not an experimental model that has been submitted or a computed model that has been submitted to a database? You can still, so if you go to the home page and under visualize, click on MolStar, it'll actually launch a standalone MolStar tool for you. So the MolStar tool here is not connected to any structure summary page. You could upload or import files. So you can open a file from your computer, you can upload a file, or you can even call on a PDB entry. So here, for example, I'm going to call on 4HHB again and click on apply. So that same thing came up. But what I'm going to do on top of this is that I'm going to also call the AlphaFold structure. And I happen to know that the UniProt ID for the alpha chain of hemoglobin is P69905. I'm going to call that. And this lighter green structure that has shown up is the AlphaFold human alpha chain, hemoglobin alpha chain, but they're not quite aligned, and I don't know what to do with this now. So, but the tool allows you to do an alignment right here. So what I can do is I can select the two alpha chains. Now, instead of being at the residue or atom level as we were doing for measurements, I'm going to go to the chain level. So if I click on any part of this lighter green, the whole chain gets selected, and the darker green, the whole chain gets selected. So I have two chains, two alpha chains, hemoglobin alpha chains that are selected. I can now say structure superposition by chains. These are the two chains that are selected, superpose, and voila, it jumps and it is superposed. At the bottom of this 3D canvas, the RMSD is reported. It's very close. But one thing I wanted to point out is that look at the N terminus here. Sorry, I have to get out of the selection mode. I'm going to click on this. Why is it not showing up? What I wanted to point out to you is that notice the AlphaFold protein starts with the UniProt residue 1, which is a methionine, but the structure, the experimental structure actually starts with the next residue, which is a valine, I believe. I don't know why this is not working. Anyway, and so this idea actually came up in one of the questions that someone had posted in the Google Doc, is that when we are looking at these models, they are actually modeling pretty much the whole sequence, including any signal peptides or residues that are removed after the protein has been expressed that may or may not coincide with what the expressed protein's experimental structure shows you. And so we should be mindful of that and be aware and pay attention to these little details, because that may have implications in where the functional sites are and how the ligands interact, etc. So I showed you how to upload coordinates. I showed you how to compare structures, and this is what we did. This is the exercise we did so far. And I'm going to shift gears to now looking at not just one or two, but large collections of structures. And to do that, I'm going to go back to our structure summary page. In the structure summary page, so I'm back in the hemoglobin structure summary page. And here, what I wanted to point out is that we did a search where we searched for all structures in the PDB that have a particular UniProt ID. But what I'm going to do here now is I'm going to pull up all those instances or all those structures that have that specific UniProt ID as not a list of structures, but as a group, as a collection. And what that entails, so I can access that either here or over here. So this is a UniProt group or UniProt group here. I'm going to click on this. And so it's telling me that there are 311 such structures where this particular UniProt appears. It tells you when these structures were released over time. It tells you whether they are all, the majority of them are X-ray structures, some electron microscopy, neutron diffraction, one computational model structure also, and it tells you other things. What is interesting is if I click on this group sequence tab, so this is like our 1D view where it is showing the sequence of all the chains, all the polymer chains that correspond to this particular UniProt ID in all these different structures. And notice it's not just one instance, all the instances are listed here. So this _1, _2 is telling you that. Some annotation gathered from other data resources are mapped here. You can click on structural features like cath and SCOP, etc., telling you where the different domains are, which parts are helical, which parts are not. Binding site. This tells you some interesting things about where all these different ligands interact. So some of these ligands interact with this region, some of these ligands interact with this region. And of course, we are showing a linear presentation, but actually this is a 3D structure. And so to truly appreciate this, what we can do is actually open that 1D, 3D alignment. And so if I click on this, so this is similar to that 1D, 3D view that we had seen earlier, but now we are not looking at one structure, we are looking at all these structures. So everything that has the UniProt ID that is appearing in any structure is listed here. The top structure, the top row shows you the AlphaFold structure. So this is already, displayed for you, but I can turn on any number of structures. So this is again showing you just the matched region, along with the whole of whatever else is in the structure, along with ligands. Okay, so I think that this gives you a very easy way to turn on, and you can turn on as many structures as you want, and look at all their ligands and all their structural, whatever other chains that are present in here. So what this also offers you is if you scroll down, notice here there are these little pink blobs. These pink blobs are indicating mutations. So if I click on this and zoom in here, so I'm going to hide some of these for clarity. So the AlphaFold structure has if you can see at the bottom, so as I mouse over, the alpha fold structure so this is valine and this is glutamate. So there is a mutation, and this is what the result of the mutation would be. So all these structures are superimposed. And I can scroll down in this list and see that there are other instances where there are mutations here, and what those mutations are, how they compare to this other mutation that I was displaying. All this is possible just on one screen by turning on and off these different buttons. And I think this is a very powerful way to look at trends and patterns and see how things are changing in the face of whatever structures you're looking at. So with this introduction, I'm actually going to shift gears. So this is what I showed you. I'm going to shift gears towards this Arabidopsis thaliana resistosome story. So we already saw in our earlier before break talk how we can find structures of this Arabidopsis resistosome. And we came upon this structure earlier. But I wanted to actually start here and ask some of these questions. So this looks like a pretty complicated assembly. Which one of these is Arabidopsis is this resistosome protein? What does this protein interact with? How can I figure that out? What ligands does it interact with and where? And can I learn something about mutations? How do I do other analysis? So I'm going to quickly go through this. And I know I'm running out of time, I hope. So the PDB ID is 6J5T. 6J5T. So to quickly scroll through the page and look here, I can see that the third entity with these chain IDs is the resistosome protein. And if I look at, so you can look at the 1D, 3D and look at the whole structure, but I actually wanted to open, or let's do that quickly, just so that. So this is the structure that I showed on the slide, but I can quickly click on this to see where the interactions are with this particular ligand. This is where the ligand is bound. I can look at various mutagenesis points and notice where the ligand binds, there are mutagenesis points. So clearly mutations in those residues would interfere with the protein's function. And as I mouse over this, notice on the top right, it tells you how there is increased sensitivity over here, if you notice. There is increased sensitivity to such and such tissues if there is a mutation. So things like that. So you can gather all this information. What I can also do is for that, if I pull up that whole group thing and look at that 1D, 3D alignment. So this is again the alpha fold structure for that protein, but I can open up. This particular structure looks very similar to the alpha fold protein. This part is missing, that's a different matter. But if I look at this structure, it's not as well aligned. And so that's surprising because it's the same structure, same sequence, same protein. And if you remember from the overview slides that Dennis had presented, there is some conformational change going on. And in order to accommodate the conformation, so this particular algorithm for superposition does not give you a good sense of that. But what I can do is I can go to pairwise alignment and I can say, I want to look at 6J5T and I'm going to look at this alpha fold. Sorry. I'm going to just copy it from the document because I believe I have put it in there. This one. So let's go back to this. And instead of doing the default, I actually want to do the FATCAT flexible comparison. And so the default was not working as well as we wanted it, but now this actually does work much better. And oh, I aligned the wrong chains, but you get the idea that I'm trying to say that, you know, you can use a tool like this to inspect and analyze which chains are relevant to your topic of research. And then you can use the other tools that we offer, like this pairwise alignment, using the different options that are available to get a better match or a better understanding of what is going on. So that is pretty much what I wanted to cover. In the notes, I've listed all the tools that I had used. So we looked at the single structure analysis, we looked at a standalone mole star, and we looked at how viewing groups of structures and their 1D, 3D alignment can give you information. With that, I will stop, and I apologize for taking longer than allotted to me.
Thank you very much, Shuchi. Are there any questions outstanding in either the Google Doc or the Q&A or the chat that we need to address?
I think there's a few of them answered so far.
Yes. Okay. Very good. And Shuchi, I think that there's some addition. There may be some additional questions for you in the in the Google Doc.
I will.
So thank you very much. So let's now turn the floor over to Qizhi Zhang, who's a member of the KBase team at Argonne National Laboratory. And her title is KBase Apps for Protein Structure Data Communication and Integration with RCSB Protein Data Bank. Thank you very much, Qizhi. Floor is yours.
Thank you, Stephen. Thank you for the introduction. Let me share my screen. And I hope you can hear me and also see my slide. Loading.
You are good to go.
Thank you. So the title is, Stephen already introduced, I just skipped to my, up to today as the afternoon progresses, I have learned a lot. I don't know about the audience. I hope you guys also learned. I am a software developer with the KBase at Argonne National Lab. So I'm looking things mostly from programmatic angle. So far we have learned that RCSB PDB has not only a vast amount of data, so rich, 1 million CSM structures and 200 experimental structures. And not only that, but also behind it, I dug into many of the data, the annotation data included. And now we learned not only those data, but also so many tools, what Dennis and Brenda and Shuchi just presented. On the other hand, my colleagues, my boss, Chris, and my colleague, Janica, also presented from the scientific front that KBase also has its own strength, especially the genomic data. Not only we have reference genome reference data. I know that because I loaded many of those data into KBase, but also we have users specifically isolates and the individual user, they can upload their data into our KBase system. So now we all know the potential now. I'm so lucky my presentation is at this spot. Everybody, thank you, the previous speakers, you have paved the road for me. So my job is going to be very simple. I'm going to just introduce to you 4 KBase apps that will kind of bridge our KBase's function and the data and take advantage of the vast amount of rich data from RCSB database. So here are the 4 apps we have developed for this specific course and also for this purpose. The first one is we want to import. And we already, I should say, we have a data type in KBase set up. We name it called the protein structures. So it's a data type or data object, as Chris said. We have KBase save data into objects instead of files. But this app, I will take a CSV file as a metadata file to define what the data the user wants to link his or her KBase data with the RCSB structures. So that's the first app. Second one, as Dennis mentioned, when you do the just simply the Google-like search in RCSB, you get the results. If you click on a JSON button, you get to a web interface of the JSON query builder, and you get the result. You run it against the RCSB database. And he also mentioned that this app is actually built on that interface, except it's programmatically from inside the KBase. The third app is PDB import metadata into a whole KBase genome. This one I'm going to just put into here because it's in a bunch of our apps, but Chris will dive much more deeply into it. The last one is after I could link, I could first search the RCSB, find the hits of matching to my interested enzyme or protein, and then I wanted to make the connection and save those data and the structure into KBase. So this is the last. I have my demo in this KBase narrative, so you can click on it and get into there. But before we get into there, I want to roughly go over the purpose of each app and the input and output look like. So the first app is basically just if the user has his or her own structure files already, like I did something for the GSP, I ran the AlphaFold tool to get the structures, PDB files. I should have asked for CIF files at one time. And then I also have a colleague who researched the M-Lotus genome and loaded them into KBase and have the genes and the functions started the info. So I know I could use this as an example to relate the AlphaFold structure with the KBase genome data. So this is one of the example input file I put in here. At the bottom, I align it with the highlight. It's actually a CSV, comma separated value file. That's a text file. The first column is the narrative ID. So first, this is a KBase term. You have to have a KBase account, and then you can create all kinds of narratives. You can see the narratives other users shared with you or public, like today's narratives. So go back to the previous slide. See, this one is my today's demo narrative. The last number is the narrative ID. That is inside the KBase, we use an ID to identify. We could use name. We have actually narrative name to But in this app, I require the narrative ID where your genome objects is existing. And also you can specify inside of that genome which genes you want to look at to associate with which of your structure file. The last column will be specify, is it a model? And then you put into this input, and then the interface inside of a KBase narrative is very simple. You just upload this file, which you already uploaded into the narratives by import. Remember, Chris was showing you can upload your data into KBase staging area, not only this input file, but also all these structure files you needed to load into the staging area in order to run this app. So that's the purpose, is eventually if you find a certain similarity identity threshold, if that's met, then you want to upload this into, I said earlier, protein structures data object. So the second one is to programmatically install the interface Dennis was showing, the query. But we also in the Google Doc, people are asking what kind of others you can search based on the advanced search features Brenda was showing, actually so rich. You can search pretty much by anything because it's a Google-like search query on the top. But I have currently installed is you can search by sequence, by protein sequence, by UniProt ID, EC number, and you can use your chemical INCHI code, SMILES code, and maybe some more, but notice here you can add multiple search them. Each one could be fixed with a logic and or, but it's built in. This is something I'm still learning. So here I put one as a logic and operator for you to check, is because I know that each category you can do and or, but inside individual ones for UniProt IDs, for example, if you have two UniProt IDs, you will be just the AND logical way. But for EC numbers, it's just OR. Okay, I jump to the next one now. And at the bottom, you can specify your E-value cutoff threshold and also the similarity, sequence similarity cutoff. And then you can run a search, you get the result. Similar to Brenda was presenting, but this is all inside of KBase narrative. The third one, as I mentioned, and Chris will dive into in more details. The fourth one is to link the two features, two apps behind the scene. Now I can find this PDB hits of what my interest, the search criteria is gave to me. And I also have my own genome data inside the KBase in the narrative. I know it's narrative ID and genome names and the feature IDs, so I can use this app to upload them into the protein structures, which will hold annotation metadata from the structure itself provided by RCSP, and also from our own KBase genomes data together. So this is, you can, one individual input, you can have multiple. So this one will be the second one. So you found your hit, this one first, the second, and so on and so forth. And then you can upload them. But you have to also specify a threshold for the upload. So now we're going to the demo mode. So here is the, yeah. As you can see on the top, I have my narrative. Oh, if I do this, I cannot show, but you can see that the URL carries my narrative ID for this demo narrative. And I have to, before the presentation, upload the data into, let me see, into my staging area. Let me exit out first. But if you wanted to, because this, I cannot exit out. If you wanted to follow along, because this narrative is public, you may want to preload those data into. So I put that link at the top of the, so where can I go back? This is what Shuchi was smart to just not use a full screen. How did I get back this out? All right, let me go to full. Stop. Oh, can I stop share and reshare? Because my full screen don't allow me to click.
Of course,
Yeah.
you should. Do whatever you need.
Yeah, so sorry about that. Yeah. Let's see. Oh, suddenly lost that. Sorry. Now I'm coming back. I linked out to that and then lost that work. Okay. So I'm not clicking on that now. I'm just go ahead to do the demo. As I showed earlier, the first one, it's very simple as long as you have the metadata file already uploaded into the staging area. And then this is the input UI. You go here. It's still loading. So if you go here, it will go into your staging area, grab the input. Now it's coming up. So these are the files, including your PDB files and CIF. I have one CIF files uploaded into your staging area, which is inside of the narrative. Click. You have an import area. And then if you have on your, it's weird, I'm in my Argonne office on the internet. So you just drag and drop from your local computer, your PDB files and your metadata file put here, it will be uploaded. This is you call it your staging area. So and then in your app, you can select that one. And I already pre-run this app, so it will generate the result like this. In the file, in my presentation, I was putting 5 different PDB files and with one genome and 5 different genes. And one of them didn't match my criteria. So it just said that one of those actually was a Rosetta fold. I have a Beck's genome CIF. I just put here, I knew didn't have anything to do with these genes, just to show if it's under the threshold, it won't be loaded. And then here, from here, you can see in this output, I can link out to see the landing page of a KBase of my specific genome. Hope this won't drop me out. Okay, so, okay, and then this one will show the most star. If you mouse over it, we'll see the structure of the most star. And I can also open a bigger window. By the time I think of my landing page. Oh, how come my landing page go to this data view of my first data? All right. I'll try that one. So and then we can see the most star here with a bigger window. And this will show the tabs. You can go through individually, and each one of them can have a and then you can also have all of them showing in one most star presentation, and all the features down here. You can do your measurement, you see your components. If there is a ligand, you can see your polymer, you turn it off and on. But this is all my 4 structures in one place. If you click on this tree here, and then you can show, maybe you can see, I don't want to see the first 3, I just want to see the last one. Then you get to see one of them, right? Yeah. And then you can see 2 of them. Maybe this is the time we just learned from Suchi that you can do some superimpose of the structures. In this case, they are all very similar, so you can color them in the regions as well. But this is to get our uploaded the KBase data together with the structures annotation data into. I don't know why. Okay, the data view didn't show somehow database KBase. All right, so that's the first app. Second one is for querying that I already showed. You can put a different filters put in there, and then this is the result. I have a different result report table here. So we get, in this case, I get how many 8 hits because of the criteria I set it here is 0.35 for the identity cutoff. And then the results would be 6, 7, 8, depending on that. Sometimes you can get more, but you can narrow it down. And then here shows the score of the return from RCSP. I just learned that today you guys use the MMSeq method. And in my previous, the first app, we use BLAST to do the match. And here we have the columns showing the strand of each, what are the chains that match the cutoff threshold, and the other links, including the publications found. These are all from the data from RCSP. But we also do the matching of the components and match the CPD, the compound ID, and then that will link out to our model seed biochemistry information. And the others, UniProt and UniProt EC numbers all can link out. Of course, what Shuchi was talking about, the 3D view of these, because these are all found inside the RCSP, so I just thought automatically link it out. It's something to do with the, okay. It's my computer today is so slow. Okay, that's the second app demo. And then the third one is, so after we already found so many interested hits in RCSP, and I wanted to upload them into KBase. Here's the example of the upload, I call it import RCSP structure. Let's show the result. Here's, this one is just to show the interface UI. All right, this will be similar to the previous one, but also different. This time, these are all RCSP entries, and these are KBase's genome genes as before, and we matched the chains and with the sequence identity calculated using BLAST. And we found it because I know I ran many times, so I set the cutoff at 68, so we get all these hits. If you set lower, then this number will be a little higher. And likewise, this will link out to the RCSP 3D view, and this will link out to the KBase landing page. And then we can explore the KBase's genomes data structure and properties. I don't know why it always get me to that. Welcome to KBase. Let me see, try one more time. Goes to the home. Oh, because I didn't sign in. That's okay. Now I can see if I sign in, I should be able to see my own data. And with that, yeah. I'll get this one is my ID. So anyway, this is 2, 1, right? Data view. It's just so that, oh, it's already showing. So you didn't sign in, and KBase will welcome you. After already sign in, it's not. Anyway, so these are the apps I built to integrate the data with the RCSP from KBase. And there's one and running out of time, so I will leave that one for Chris to talk about. And if you have any questions and comments, please leave into the Google Doc. I'll go over. I actually wanted to hear more because this is the first version of the app, so we come up with. So there may be more requirements and more suggestions that are all welcome. With that, thank you very much. Sorry for the.
Thank you very much, Kizzy. There's no reason to or need to apologize.
I forgot to sign in. I thought I could see my narrative. I already signed in.
So our cleanup batter will be Chris Henry again, who led off. And he's going to provide a little bit more color on how KBase and the RCSP PDB have been collaborating together. And the title of his presentation is Making the Best Use of Protein Structure Data in KBase and PDB. Chris, over to you.
All right. I think that the title should have been Cleanup Batter.
Well, it's
accurately reflects
the world's unfortunately the World Series has just ended, so you've missed your calling.
That actually reflects very perfectly. Stephen saw right through me, and that's really the purpose of my talk, to adaptively respond to anything I thought that showed up in the workshop. So I don't necessarily have 30 full minutes of material here, but I also want to give people a chance to ask questions and make comments and feedback, especially. But I'm going to highlight a number of things and also talk about some future plans here in this talk. So just to kind of give an overview.
Are you going to go into presentation mode, Chris?
Oh, yes, I will.
Great.
Just kind of give an overview.
You're good to go.
Okay. Just to overview some of the things we've covered. So we've shown how you can use the gene function discovery pipelines in KBase to identify candidate genes for functions of interest. Candidates could be proposed from gap filling or pathway predictions, annotation apps, alignments, blasts, and many other algorithms. And you can pool now the closest experimental structures associated with these genes from PDB using two different apps: our genome-wide query app, which gives you a wide but shallow pool from PDB, and our query app, which gives us our gene-specific query app, which gives us a deep query from PDB with more hits. You can export gene candidates as FASTA, generate structures in Google Colab. Again, if you have questions, like QZ is the one that did this, so she can, if you have questions on how to do that, she can give you some information. Then you can import those computational structures back into KBase or query and analyze those structures directly in PDB. And I'm going to show you something in a minute that's kind of connected to why we have these in both places. But you can use PDB query and import apps to pull structures with desired functions or substrates co-crystallize into KBase. You can query based off of UniProt or sequence, or compound structure for the ligand that's co-crystallized. So you can query and EC numbers. You can very rich query, and just encourage you to look at that app. And in particular, look at the PDB query, because the tools are very, very good in the PDB site itself, and quite capable of doing a lot. And we only offer a subset at the moment in KBase, but I hope to keep building that out. You can align computational experimental structures and in the PDB site. So kind of, and this is the big thing, like the thing I envision is you'll find structures of interest, you'll get the sequences, you'll fold them, you'll put them in KBase just for documentation, but then you take them to the PDB site and you do these alignments. And then the idea is you would screenshot these and then put these in your KBase narrative to help document the science you're trying to do. So the KBase is, in a lot of ways, it's an analysis pipeline, it's a data compilation and sharing pipeline, but it's also a publication pipeline. Like, the narratives are designed to be a tool to share your ideas with the research community. And that's a way to do that. Otherwise, it's hard to make these stories and have the data there and every step that you did captured in the story. That's why we call them narratives. So comparing PDB complexes and co-crystallized ligands with complexes and reaction substrates and models. I'll get to this and more in a moment. I'll actually demonstrate this. So the idea is it's one of the great values of PDB, particularly the experimental structures, as Stephen and others have mentioned today, is the co-crystallization information. It provides a wonderful context on the models that you build in KBase, which are based often on homology. But so that PDB is like an independent source of experimental validation for annotations that you make in KBase. And what's nice is even when our leap to PDB is based off of homology, what AlphaFold now lets you do is go a step farther. You can generate an AlphaFold structure for your sequences in KBase, many of which will be totally new and never seen before because they're from an isolate or they're from an environment. And then not only see if that sequence is homologous by sequence to a gene in PDB or in other resources, but actually see if it's homologous in structure and when there is variation, where is the variation? This is the lovely presentation that Shuchi showed, is how you can see the sequence variation and where it lands in the structure using that 1D, 3D tool. So all of this pieces together to give you a really rich readout that's so much farther beyond, it's the E-value is good enough, so let's just propagate the function. And that's the thing, like that's why we're integrating KBase and PDB. That's the thing we want to empower people to do. So you want to, and of course you can apply the import apps to look at structure coverage of a genome. And I'll show you how to do that in a second. Identify gaps in the structure coverage, which is important as well. I'm going to try to drag this. Test for existing and completeness of protein complexes. I'll show you how to do that. And then import PDB and UniProt data into the annotation of the genome. So let's before I get to the future plans, because there's a lot still to do, I'm just going to jump out to the narratives and show some of these things quickly, because not all of it's totally obvious from what we've presented so far. So first of all, when you run this app, the PDB import PDB metadata in the KBase genome app, as you recall, this is the broad but shallow. So this queries every gene in your genome with against PDB and gives you all the hits. It doesn't just bring up this nifty table, which lets you search for the hits that you're interested in, and also link out. So for example, linking out to the PDB hit for any gene in your genome is wonderful, because now you get that structure context, and you remember you tie in through this link in the PDB, you're instantly tying the gene in your genome into all of that stuff that Shuchi and Brenda and Dennis showed, like wonderful tools to look at sequence alignment, structural homology, see how sequence variation relates to structure. All of that is at your fingertips because you now have this link for every gene in your genome that has any homology against PDB. So that's the whole idea of this app, is put all of that, make all of that easy. And like something we'll be thinking about for the future is, for example, can we inject the sequence of this gene into that sequence alignment that Shuchi was showing, so that you would actually see your gene show up in that as one of the sequences in that alignment. So literally you could go to this structure page and instantly see where the variation of your gene lands on this structure. So that's something we're going to be thinking about for the future. I think it's particularly possible now that you have tools like AlphaFold that can rapidly make structures. And we're going to be looking at putting some variation of AlphaFold into KBase itself. So I think there's definitely there are many of these tools emerging now, and some variation that we should be able to put in. And just a reminder, this shows the method, all of the strands, the identity, and the e-values. This can help you craft your other query, like with the deep query tool. You can decide, I want a lower identity than this. I want a lower e-value than this. I want to get a wider set of hits. And of course, the taxonomy, and this tells you whether or not RCSB and UniProt agree about the EC number. And that is also useful information. and of course the hit to UniProt. And the papers themselves, as Jonica showed, can be quite valuable because the title of the paper can be the function of the gene you're looking at. But to get it structural, that one thing I want to emphasize, we don't just put the data in this nifty table. We also inject the data into the KBase genome object itself. So the data is not just there for the user to see, it's injected programmatically into the KBase data model in the form of these ontology terms. So these are the EC numbers that were added. And what this is saying is that this is the EC number, and it's saying this EC number was because this gene is a hit for this structure in UniProt. And this is for UniProt. This EC number is in here because this gene is a hit in PDB. So it shows you where the EC number came from. It also sticks the co-crystallization information in here in the form of the model seed compound IDs. And as QZ showed, if you click on these compound IDs, it takes you to the model seed site with a picture of the structure and all the compound information and any reactions that compound is involved in. So you've got those links, not just in this table, but also in these ontology terms and you've got the RCSB IDs themselves. One of the nice things the RCSBs give you is if you search in the genome for the letters RCSB, it will filter out to only the genes in your genome that have a hit in RCSB. So this gives you a very quick readout on the structural coverage of your genome. So this gives you 1,730 genes in your genome out of 14,000, but this includes all look for the word gene out of 4,700. So roughly a third of this genome has a hit in PDB. And so for Arabidopsis, you can do the same thing. I think I have that. Yeah, here. So it's 8535 out of about 14,000. So I actually, interestingly, for the plant, over half the genome has a hit in PDB. Interesting with the coverage on the genes for the structural information. So I'm just, as cleanup batter, I've got notes on things I wanted to hit. The other interesting thing is complex completion. Because we have the structure information stashed in the genome, I plan on connecting this structure information to the modeling, because the metabolic models have definitions of the gene complexes that form the enzymes that make each reaction happen. So we can compare those complexes to the complex information from PDB. And you can see this kind of exemplified perfectly if you look up something like ATP synthase. ATP synthase is a classic example of a gene that has a ton of subunits and the complex. And so by querying for this in this PDB query tool, we can ask questions like, does my luteus genome, is this complex complete here? So if we're going to annotate a complex to a gene in a genome, one question to ask yourself is do I have evidence that this complex is actually completely represented in my genome? So for this case, you can see one gene hits the first 3 chains, another gene hits the next 3, and another gene hits the final chain. So yes, this is and they're all homolog, they're all next to one another on a chromosome. So it's just a really nice demonstration of how complex information can be another source of validation for annotation, but also evaluating whether or not my genome itself, is my genome complete, are the complexes complete. And again, this can be done manually using the views that we offer you now and querying in clever ways. But I'm working on one of the next steps is to figure out how to do this programmatically, so that you can just with the click of a button instantly get a readout on how many of the complexes in your genome are complete versus appear to have missing steps. So it's sort of a complex-based gap filling that we'll be able to power in the future with this integration with PDB. On a similar note, the components is another interesting thing, because you see here, this is ATP synthase, and of course you're seeing components you would expect to see. You're seeing phosphate, you're seeing ADP. Obviously, these are things that these are substrates to this reaction. You expect to see them in the co-crystallization, but you're also seeing coenzyme type ligands here. So like magnesium, paradoxal phosphate is everywhere. And that this doesn't mean it's a substrate. This is, in this case, these are PLP enzymes that just rely on paradoxal phosphate as a coenzyme. So the co-crystallization information that comes in from PDB and is embedded into our annotations now here is actually providing you insights about the needs of these enzymes. And that's useful again for modeling, because if you knock out PLP biosynthesis or knock it down, you're not just going to impact, an enzyme that uses PLP as a substrate directly. You're going to impact any enzyme that relies on this compound as a coenzyme. So having this rich information instantly at your fingertips, annotated into these genomes, and not just reference genomes, which we could just pre-compute all of that. But one of the big things about KBase is we're not just a reference database. We're working with your completely novel data that nobody's seen before. And so being able to rapidly and instantly query, fill these genomes out with this kinds of information is incredibly valuable. So it's interesting at the surface, but there's a lot of things under the surface. And I'm thinking about, for the third-party developers among you, know that you have this data at your fingertips to do stuff with. You can, tons of integrations that are powered by having this data available. I think that's the last thing I wanted to mention is the punchline of the Arabidopsis story. So the punchline is what you see from the study that Brenda did with the alignment, is that the two proteins in Arabidopsis actually align to two different parts of the E. coli gene that does with the RIBD function. And so one of the reasons that this function was mysterious for so long was that the most obvious gene that was the best homology hit, I think, which was 47390, you could stick it in E. coli and it would not do RIBD on its own. It did not fulfill the function. You actually had to put both genes in. So both genes are required. And now with that knowledge known in our current annotation of Arabidopsis, we now have these annotated. Both of these genes are annotated with this function. But the structure alignment actually shows you this really nicely. If you align both Arabidopsis structures to that E. coli gene, you get different parts of the structures aligning. And you get a whole other domain, which is a cog that still has an unknown function today in E. coli, but clearly has something to do with riboflavin biosynthesis. So there's a mystery for you that plant proteins are interesting because they can provide insights back into microbial functions because of the degree of fusion of functional domains and that happens within plant proteins. So yet another application here, and another value of having the Arabidopsis structures now available in PDB, as they've done, as Stephen and the team have done. That now that those plant structures are there from AlphaFold, you have that resource to go back and do those alignments to microbial genes and learn new functionality information, not just about plant genes, but about microbes as well. I think that hits. The only other thing I was going to mention was, if we go to QZ's narrative, see if I can find the right app here. Yes. So I'm going back here, and it's true that there's not a lot you can do at the moment with the structures and KBase. I think that will change soon. One of the things we can add, for example, is the structure homology search to PDB. They already have that functionality, so we can just dial into that through the APIs. So I think this will be a functionality that grows. But another point I'd make here is a value of putting your structures in the KBase using this app is that it loads the structures, it connects it directly to your genomic information. That's why we ask for all the gene ID stuff. That's why we ask for this. It's connected explicitly to your genomic information, which is also here in your narrative. And so what I'd encourage people to do is put your structures in KBase so it's shared when you go to publish your narrative with your paper. It becomes a part of your publication, and anybody who wants to get that structure later can easily download it from KBase, just using the download controls here. And then when you do things like that structure alignment in PDB with the PDB alignment tools, screenshot the results of that and stick that in KBase in a markdown cell, much as we've done with this beautiful, RCSB logo. Actually screenshot that into a markdown cell in KBase so they can see that result as well. And now you're telling your entire scientific story in a narrative format that can sometimes be easier to digest than a journal, which is often highly filtered and cut down. Having the full logic laid out someplace. That's a big part of what KBase is about and what this interface is built for. With that, I'm going to go back to my slides. Sorry for the stream of consciousness here. So future plans for the KBase PDB apps. Stephen is going to talk about some general future plans as well. I'm thinking specifically about the future of these KBase apps. So we currently, the Molestar viewer in KBase is currently only available on upload and specific upload of computational structures. But we're adding a widget so that'll be available anytime. So remember, the structures get stashed as the structure, protein structures data type, and these narratives, like these protein structure data types, they're stashed in there. And so we're just going to make it so that if you click on this data type, the Molestar view would come up. At the moment, the viewer is very minimal, but we'll make it so that Molestar view actually comes up with your structures. So that's something we didn't quite get to in time for the workshop. We want to enable painting of other data in KBase on the structural views, like sequence alignments and variation, much like what Shuchi showed. Probably less of a priority because I think the bigger priority is making sure that this kind of data in KBase can just be exported to the beautiful views they already have in the PDB site. So, however we end up doing it, that's something that's on our mind. Let's get all that data, make it so that you can easily visualize all that data without too much pain, and figuring out how to do it manually. And then integrating more, just generally integrating more Molestar functionality into KBase. And you saw all the beautiful things that Molestar can do in Shuchi's presentation. The PDB query app, we want to enable query, like currently you can only paste in sequences. We want to enable query by feature IDs in KBase. Also querying compound structures by model seed or IDs or PubChem IDs, for example, instead of just NSheet. And saving query output as a sequence set so that you can connect it to other things. You can connect that query output to, for example, the structure import app. And then enabling query by structure alignment, which is, again, I mentioned that already. And then for structure import, like expanding the import to include the computational structures in PDB and other databases. So currently this tool only supports the experimental structures. And I would generally say this query app and the genome annotation app, that only queries experimental structures. And probably always, the query apps and the genome annotation probably always will just be focused on experimental structures, because to me, that's the valuable information we want to map onto the genomics data in KBase. And then for docking, we do have an AutoDoc Mina app in KBase, but it's linked to our older structured data types. So we'll be updating that to link to the new data type and enable you to save and look at docked poses later in tools like Molestar. More broadly, we want to link to modeling, so automatically compare model complexes with the PDB data, compare model reaction substrates with co-crystallized ligands, and be able to annotate models with coenzyme data, which if you're a modeler, like people are increasingly using this coenzyme data and metabolic modeling to better capture the behavior of these enzymes. So but when people do this, it's literally a year of manual annotation effort to get this data in. So having a tool that would automatically import this data from PDB would be enormously valuable. Structure alignment, we want to get this, examples of tools that are available in PDB, or the Oak Ridge National Lab team is adding some tools along this line. And then experimental and computational structures, automatically linking these things so that you can compare your AlphaFold structures quickly to the experimental structures in PDB. As Stephen said, the computational structures really don't stand alone. You need the PDB, you need the experimental structures too, to make the best use, like pulling in the co-crystallization and really truth checking your folds. And then adding one or more computational structure pipelines. And we'd love to have any other ideas, and I'll acknowledge our many collaborators and the PDB and the KBase team, and of course all the people who contributed to our workshop at the beginning of this project 2 years ago. And I will stop there and take any questions or comments. And Stephen's going to bring up a survey, and I just really encourage you all to fill out that survey so that we can get some feedback with your questions, your feedback on the workshop itself, and also your feedback on what we built. You have a chance to get us to build different things and change these tools in the way you'd like. So take advantage of it.
Thank you very much, Chris. I'm not seeing anything in the Q&A or the chat for you. And the SurveyMonkey link has been put into the chat for those of you who want to grab it from there. But let me just share my screen, if I may, and bring this very successful afternoon to a close, and say a few words about where we're going, and also about how we got here. That's actually quite important, as you'll realize in a moment. So what you heard from Chris is some quite detailed plans in terms of further integration of PDB data into KBase. I don't have as an extensive laundry list as Chris did, but we are very interested in bringing in more annotations pertaining to both experimental structures and computed structure models from KBase, and ensuring that users of rcsb.org can easily link out to KBase when they need access to information that we're not going to integrate with our website. So I won't say anything more about plans. These are still in development, and we will be looking to our benefactors, our funders at the Department of Energy, for additional funding to continue supporting this very productive and very effective collaboration. All feedback is good, and we are very keen to get your feedback on the crash course. A series of questions have been presented to you in this SurveyMonkey. Anybody who fills out the SurveyMonkey will receive the link to the recording of the event, and also PDFs of the slides that were shown here. If you are using the rcsb.org website and you want to provide us feedback from there, you can simply go to the top right-hand corner of any web page on the rcsb.org Research Focus web portal and click on the Contact Us button. Type in your message, and one of our team members will follow up with you within a matter of days. Usually, it's within 24 hours, unless of course you provide the feedback on the weekend. So please do fill out the course exit survey, and if so moved as you're using rcsb.org, don't hesitate to contact us and help us get better at serving you, the customers. I don't want to poach any of the KBase staff, so let me be clear about that. But if there are individuals in the audience who are interested in working for the RCSB Protein Data Bank, we have a number of openings, both at Rutgers and at UCSD. They're listed here towards the bottom of the slide, and they range all the way from senior appointments to summer undergraduates and gap year opportunities between undergrad and medical school, or undergrad and graduate school. And you can find all the details on our jobs page, which is www.rcsb.org/pages/jobs. It's a fun place to work. It's a very, our bi-coastal teams are very well integrated with one another, and there's plenty of scope for publication and intellectual and professional growth. I want to advertise the last crash course of our season, of our fall 2022 season, which will be coming up on the afternoon of December the 7th. It's a Wednesday. Our focus is going to be cancer immunology and metabolism, a completely different topic, but a very interesting and important one. The nexus of cancer metabolism and the way the immune system responds to cancers is very, very interesting. Registration is required. It is free. Simply go to the registration link provided on this, either on this page or if you go to iqb.rutgers.edu, you can see there's a tab with access to the crash courses, and you can find the full information for the event on that particular web page. In addition to the speakers who gave excellent accounts of themselves today, there are people behind the scenes who help make this possible, and I want to acknowledge particularly for this event Michelle Sanguera, Maria Voigt, and Claudia Lerma Ortiz from the KBase team. And from the Institute for Quantitative Biomedicine and RCSB Protein Data Bank, I provide the names of 5 individuals who help ensure that all of our infrastructure, whether it's physical or IT, is working and supporting not only the needs of rcsb.org and the RCSB PDB, our websites, and the other members of the Institute for Quantitative Biomedicine, but of course all of our customers who visit rcsb.org or our education and outreach website, pdb101.rcsb.org. This is a huge team effort, as you can see from this slide of all the individuals who contribute to the RCSB Protein Data Bank. I want to draw particular attention to John Westbrook. It was John and Chris who launched this collaboration, who persuaded the Department of Energy to fund it, and tragically, John passed away on the 18th of October last year, completely unexpectedly, leaving a hole in the collaboration and a hole in the organization. I'm very grateful to everybody at KBase and everybody at rcsb.org who stepped up to help overcome the tremendous loss that we experienced when John passed away. He's sorely missed literally every day here at RCSB Protein Data Bank. We are generously funded by the National Science Foundation, the National Institutes of Health, and the United States Department of Energy. We are very grateful to our hosts here at Rutgers, UC San Diego, and the University of California, San Francisco. And we operate as a member of the Worldwide Protein Data Bank with partners in Europe and Asia, and also partners who run the Biological Magnetic Resonance Bank and the Electron Microscopy Data Bank. So with that, I'm going to stop sharing. And thank you once again for all of your contributions today. It's a pleasure to be able to host so many people. We had more than 100 participants online this afternoon. And you'll be hearing, now that we have your email addresses, you'll be hearing from us again in terms of other crash course opportunities that we have here at the Institute for Quantitative Biomedicine. We also run a seminar series every Wednesday at noon, with the exception of the first Wednesday of the month, and of course the summer months where we don't have regular seminars. A wide range of interesting topics, and feel free to drop in virtually and enjoy our seminars. So, Chris, I thank you for your partnership. It's been great fun working with you and all the members of your team. And I once again thank all of today's speakers for a terrific outcome.
Yes, indeed. Thanks, everybody.
Yeah, thank you very much. And we're going to finish a few minutes before time. Get home safely tonight, everybody, and